A very good morning to all of you. Seeking the blessings of His Holiness Jagadguru Sri Shivratri Deshikendra Swamiji, I, Dr. Jamuna, on behalf of the organizing committee, would like to extend a very warm welcome to all of you to today's international webinar on biofilms, health aspects and control measures. I, with great reverence, acknowledge Dr. Suresh B., Honorable Pro Chancellor, Dr. Surinder Singh, Honorable Vice Chancellor, and Dr. Manjunatha B., Registrar, JSS Academy of Higher Education and Research, for their invaluable guidance, which is cherished by all of us. We are delighted to have the gracious presence of Dr. Kushalappa PA, Director Academics, and Dr. Ravisha KA, Head, Department of Water and Health, Faculty of Life Sciences on this occasion. I warmly welcome Dr. Kushalappa PA and Dr. Ravisha KA. It is a privilege that we have amongst us today our distinguished speakers, Dr. Devendra Dusne, research scientist from Nationwide Children's Hospital in Ohio, USA, Dr. Vandan Nagar, scientific officer from Food Technology Division, Baba Atomic Research Center, Mumbai, and Dr. Veda Egde, professor and head from Oral Pathology and Oral Microbiology Department, SDM College of Dental Sciences and Hospital, Dharwad. I wholeheartedly welcome all our keynote speakers for today's event. I also welcome all the honorable guests who have joined this webinar, members of the SIG, the heads of the JSS constituent colleges and various departments, my colleagues from Faculty of Life Sciences, faculty and scientists from other institutes, all the participating delegates, research scholars and students. We appreciate you taking time of your busy schedule to join us today for this event. Microbes have been studied as planktonic and freely suspended cells so far. However, the discovery that microbes attach and grow on exposed surfaces and exhibit a biofilm mode of growth has led to a paradigm shift in the way we look at microbes. Microbial biofilms incur economic losses, cost and billions due to economic damage, product contamination, equipment damage, medical infections, and also impact industrial systems and human health. The study of biofilms has led to a new way of understanding microbiology and to rethink strategies for dealing with microbes as a biofilm, an issue that in the past was overlooked as the significance of biofilms was not yet recognized. The treatment of pathogens now has to be reconsidered in the light of this new understanding of how microbes actually present themselves in our environment in the form of biofilms. So far, significant progress has been made, but there is a lot more to understand and do for developing effective treatments and in educating society about biofilms and their implications. The current pandemic has shown the relevance of investing in research and on healthcare. Our SIG on biofilms in clinical settings is committed to working on novel strategies to overcome problems of antimicrobial resistance and biofilm formation in clinically relevant microbes. We hope that by attending today's webinar, all the participants will learn a lot and will have a fruitful time as we have lined up 3 pertinent talks in today's sessions that are emphasizing on the role of biofilms in clinical settings, food safety, and oral health care. Now, to begin this program, I request Dr. Ravisha KA, Head, Department of Water and Health, Faculty of Life Sciences, to deliver the opening remarks.
Thank you very much, Dr. Jamuna. My respectful pronouns to His Holiness Jagadguru Sri Rajendra Mahaswamiji, and also I seek the blessings of His Holiness Sri Deshikendra Mahaswamiji. My respects to the Pro-Chancellor, Professor Suresh, Vice Chancellor Dr. Surinder Singh, Registrar Dr. Manjunath, and all the other officers of the university who have helped us for the conduction of this, actually formation of this SIG and also conduction of this program. I wholeheartedly welcome Dr. Kushalappa, who is the man behind all these SIG groups and who has been a great source of inspiration and encouragement for all our faculty and students for this novel idea of SIGs and taking them forward for developing some novel strategies to overcome the issues pertaining to these SIGs. Friends, as you all know, biofilm is an assemblage of microbial cells, particularly that are irreversibly associated with a surface and usually enclosed in a matrix of polysaccharide material. Biofilm is composed primarily of microbial cells and extracellular polymeric substances. And these extracellular polymeric substances plays various roles in structure, function of different biofilm communities. Adhesion to the surface provides considerable advantages such as protection against antimicrobial agents, acquisition of new genetic traits, and the nutrient availability and also metabolic cooperability. In fact, I must remember Anthony van Leeuwenhoek here, who discovered microbial attachment to his own tooth surface, who is credited with the discovery of this biofilm. The formation of biofilm, as you all know, takes place in 3 steps, and biofilm is responsible for various chronic bacterial infections, infection of medical devices, deterioration of water quality, and the contamination of food. I'm sure this webinar of the special interest group that is primarily formed with reference to looking for novel approaches of managing these biofilms to overcome a lot of health hazards and health associated disorders and diseases provides an overview of the formation of biofilm, the structure, role, and in special microbial communities and its application. The learned speakers who have been chosen by this organizing committee have done significant contributions in their own area of interest. I'm sure their expertise and experience will ignite and inspire our young minds and also give new directions to the new directions of research and also take up projects associated with this SIG group. I hope this webinar will pave way for some fruitful projects that is developed interdisciplinarily along with interinstitutional involving different institutions and will take a good share in contributing new knowledge in the area of these biofilms and their management with special reference to health science applications. I thank Dr. Devendra, Dr. Veda, and the other speaker who have accepted our invitation to be part of this SIG webinar and sparing their valuable experience, expertise, and also time for the good of the institution and also for the cause of biofilm knowledge generations. I also thank the organizing committee and the special group in particular for having thought of such a wonderful webinar to see that some of our young minds and particularly the young scientists and PG students and PhD scholars and faculty will be highly benefited. I thank one and all for the gracious presence, and I also thank all the participants who have been here to learn and interact in this webinar. The discussions and deliberations that is going to happen during this webinar will certainly pave way for new knowledge and new lines of research in the days to come. Thank you one and all. Thank you very much for providing me this opportunity to be a part of this international webinar. Thank you.
Thank you, sir, for that wonderful remarks and for highly encouraging and supporting us in all our endeavors. I would like to invite at this stage Dr. Kushalappa PA, Director Academics, to address the gathering.
Thank you, Jamuna. First of all, I seek the blessings of His Holiness Swamiji for the success of this program and for the success in all our endeavors at the JSS Academy of Higher Education and Research. And I thank the organizers, especially Jamna, for having Jamna and your team, Sindhu and the others, for having invited me to be with you all. It's my pleasure. And to tell you the truth, I did a lot of reading yesterday so that I could at least talk 3 or 4 sentences with such distinguished people like Dr. Devendra, Dr. Vandan, and Dr. Veda Hegde over here. I didn't want to make a fool of myself, so I went through a lot of literature. And being a health professional, I am fascinated that the SIG group biofilms in clinical settings and their control. You're doing a wonderful job in having the webinar biofilms, their health aspects and control measures. Biofilms is something which has been there with us from ages, and the most glaring example is the green algae which we see on the surface of any water bodies, and the clogging of the pipes. But I was more interested in the information I got regarding how they can aggravate any condition of the COPD in pulmonary conditions, the chronic obstructive pulmonary disorders, and especially cystic fibrosis, and how the biofilms can have an effect on their healing. And the nearer home are the dental plaques, which are mainly caused by the biofilms. This really took a lot of my interest, and how the biofilms are resistant to any measures to control them. And it was a fascinating subject, and I thought I must be with you all today and see what our experts can give their opinion on. And I was also fascinated to read that the same biofilms which cause so much of problems are also utilized to treat water, the sewage water, and that was something which was fascinating. And I hope I can get some insight from the experts who are here. And on behalf of the I bring you greetings, all the resource persons, renowned persons which are here, who are in the webinar today. I bring you greetings from our Pro Chancellor, the Vice Chancellor, Registrar, and other authorities of the JSS Academy of Higher Education or the JSS University. And I hope the participants will have a wonderful day of knowledge sharing. Thank you so much for inviting me, and I wish you all the best for the webinar.
Thank you, sir, for the kind words and the unstinted support that you always provide us in our research and academic activities. Thank you so much, sir.
Thank you.
Okay. Before we get started, I have a small announcement to make. If you have any questions during the presentation, please type them into the question box in your control panel, and we'll bring them up at the end of the presentations. Now, without further ado, we will turn the time over to our keynote speakers. I now request Dr. Sindhu to introduce our first speaker, Dr. Devendra Dosne.
Thank you, Jamuna. I'm Dr. Sindhu, Assistant Professor in the Department of Microbiology, Faculty of Life Sciences, and member of SIJ. It takes me an immense pride to introduce today's speaker, Dr. Devendra Dosne. Dr. Devendra Dosne obtained a doctorate in 2011 in biotechnology from the Institute of Bioinformatics and Biotechnology, University of Pune, India. His PhD was funded by a collaborative BARC and University of Pune program. After PhD, he was offered a postdoctoral fellow position at McGill University in Canada, where he carried out the pioneering work on applications of alkaloids and natural compounds from cranberry and other berry compounds to potentiate the efficacy of antibiotics, inhibit bacterial motility, and disperse bacterial biofilms. Dr. Dosne then moved to the Ohio State University in 2015 and worked as a research scientist with Professor Paul Studley. Dr. Dosne also served as a scientific consultant for one of the leading medical devices company, that is Biocomposites Limited, based in UK. His research focus is to develop novel methods to detect and strategies to inhibit bacterial biofilms associated with orthopedic and wound infections. Dr. Dosne joined the Nationwide Children's Hospital in Ohio, USA as a research scientist and focused his research on studying animal models for burn and wound infections and developed therapeutics for the treatment of these infections that could further be used in burn patients. Dr. Dosne has received several prestigious awards. Some of them to name here are Young Investigator Award from Center for Biofilm Engineering, Montana, USA, the Journal of Wound Care Award for Biofilm Research, International Society of Chemotherapy Novartis Biotechnology Leadership Camp, as well as few travel grants and recognitions. Dr. Dusani has contributed to more than 40 research articles in peer-reviewed journals. He is currently on the editorial board as a review editor for Frontiers in Microbiology and also reviewer for PLOS ONE, Clinical Orthopedic and Related Research, Biofilms, and few other microbiology-related journals. With this brief introduction, I now request Dr. Devendra Dusani to deliver his talk on biofilms from bench to bedside, sir. I welcome you, sir.
Yeah, thank you very much for the introduction. Good morning, everyone. First of all, I would like to thank the organizers for giving the opportunity and inviting me to give this talk. So I'll share my screen and start with the presentation. Are you able to see the first slide?
No, sir. Yeah, now it's visible.
Okay. So the title of my talk is Biofilms from Bench to Bedside. I've been working on biofilms for almost more than 15 years. And in this talk, I'll be focusing a part of my research on orthopedic infection and the role of biofilms in these infections as such. So the biofilm related infections, mostly if you see that it can cause the nasal colonization, oral diseases, cystic fibrosis, pneumonia, or urinary tract infections, or even the orthopedic infections as such. I've mentioned few of the causative bacteria which can form biofilms on these particular surfaces, and they are responsible for the infections as such. A very common example I can say is about the biofilms which is growing on the toothbrush. So this is just a single bristle, and you can see the biofilm forming on top of the bristle as such. This is the enhanced image of the same bristle. This is the bristle and this is the biofilm which is formed onto the surface of the bristle. The other good example which I can give is about the wound infection. So the wound infection, when it is stained with the green dye, for example, this is Pseudomonas aeruginosa, and whatever you can see in blue, these are the host cells. You can see that the green biofilm completely colonizing the wound as such. Another staining method suggests that the red biofilm is completely colonized onto the wound surface. So coming to what are biofilms? So these are the steps of biofilm formation in which the single bacteria which is free floating, this is basically called as the planktonic bacteria. And when it comes and attached onto a surface, the surface could be any implant material, biomaterial, or the host surface in case of the infection as such. So the bacteria comes, they might also aggregate by attachment to each other, like a cell to cell interaction. And there could be aggregation and attachment onto the surface. So this process takes within minutes, whereas the further second step, which is the microcolony formation, in this step, what happens is there's secretion of the exopolymeric substance that's called as the exopolysaccharide. It could be proteinic or another material, could be protein or lipid or other kind of material depending upon the causative organism as such. So this forms a glue-like structure in which there is biofilm or the cells are entrapped. This is called as a microcolony. And the microcolony usually is, the thickness is in microns. The final stage is where the maturation of biofilm takes place, where the bacteria they multiply and the biofilm thickness is increased. Also the host cells you can see here. So these host cells are observed And with this host cells, there's interaction of the biofilm. So this is a reversible process. After this, the planktonic bacteria, the single bacteria, they can emerge out, again find a suitable surface and attach. So this process goes on continuously. And once the biofilm is formed, it's very difficult to eradicate this biofilms as such. So coming to the importance of the biofilms, according to the NIH, up to 80% of the human bacterial infections are caused because of the biofilms. So only 10% of the bacteria is present in planktonic or free floating form. And it is very easy to kill these planktonic form of bacteria. However, 90% of the bacteria exist as biofilms. And because of the production of the EPS, so these biofilms are very rigid and almost 1,000 times more resistant for the antibiotic as such. Now, coming to the mechanism by which they can, the antibiotic resistance is maintained as such. So this is a cross section of the biofilm. This is the surface where the biofilm attach, and the full grown biofilm consists of the basal attached surface. So this is the top of the biofilm, and this is a planktonic bacteria as such. So when the antibiotic is coming in contact with the biofilm, there is very slow penetration, the one shown in blue. So what happens is the antibiotic is able to penetrate and kill this bacteria. However, because of the thick exopolysaccharide, it is not able to penetrate deep inside, and you can see that there are some resistant bacteria within the biofilm which emerge, and these are the ones which could cause severe infections as such. The other one is the surface attached altered microenvironment, where you can see that the zone of nutrient depletion as well as the waste product, they come in contact with the antibiotic and they neutralize the antibiotic. So because of this, the biofilm becomes very much resistant, and almost even at the dose of 1,000 times the MIC, the biofilms is very difficult to eradicate. So in my research, basically I'm looking at the understanding of how biofilm infection takes place, how the bacteria within the biofilm become resistant to antimicrobial treatment, and to develop novel strategies to treat these chronic infections. So I'm understanding the cell to cell, that is the bacterial cell to cell and cell surface interactions during the biofilm formation, the interaction of the pathogen and the biomaterial, as well as the interaction of the pathogen and the host cell. So mostly I'm focusing in this talk about the role of biofilms and how it causes the orthopedic infections. Considering a classical example of the knee, so here you can see that this is a biofilm which is formed on the knee as such. This is a primary revision. Basically what happens is after the knee surgery, they place the metal or other component as such at that particular place, and these surfaces or implant surfaces are very much prone for the bacterial infection as such. So giving you more in detail about what exactly happens during this infection. So the process can take place in 4 steps. The bacteria, which is the planktonic bacteria as such, they can come and attach to the implant surface, adhere to the surface, and form these thick exopolysaccharide and biofilm which is attached onto the implant. So I'll talk in detail about the bacterial cell as well as the implant interaction. The second type of infection which is caused is due to the bacterial aggregation. So what happens is because of the presence of joint fluid, it is also called the synovial fluid. So synovial fluid has some components by which the bacteria can form aggregate, and these are the aggregate which are present. So the third mode of infection is these are the host cells. So the bacteria can come in contact with the host cell, penetrate, and grow intracellularly. So whenever there is antibiotic, added to these kind of infections, the antibiotic is not able to go inside the host cells and kill the intracellular bacteria. So that's why they can evade the host system as such. Another mechanism is the host neutrophil and bacterial interaction. So neutrophil are the first line of defense mechanism by the body. And what happens is because of these neutrophils, they engulf the planktonic bacteria, but the biofilms are very difficult to engulf. So I'll talk in detail about each and every step. So coming first to the bacterial cell and the interaction of what happens. So these are the planktonic cells, as I said earlier. They come in contact with different implant materials. They attach and form biofilm. And this biofilm further matures, and the planktonic form of bacteria again is comes in contact with another surfaces, and colonization takes place. So in this step, what happens is there are different detection techniques which we use in the laboratory. This is a classical example of one of the implant material which we obtained from the orthopedic uh, infected patient as such. So this was taken, it was sterilized, autoclaved, and in the laboratory, we wanted to see whether the surface, whether the smooth surface or the curved surface, which is more prone to infection or biofilm formation as such. So what we did was we grew we took this metal surface, which was sterilized, and this metal was inoculated with Pseudomonas aeruginosa. And after 2 days, we removed that, washed with sterile PBS, and overlaid with the agar. So after 24 hours of incubation, we observed that these specific crevices, these are more prone to formation of the biofilm as such. Since this Pseudomonas, what we used, it is a bioluminescent strain. We took this and put it under the IVIS imaging system and observed that the rim of this particular implant as such had more biofilm as compared to the other surfaces. So based on this, we can say that the smooth surfaces, there is hardly any biofilm formation, whereas if the surface is rough, it has more of the biofilm as such. The other technique what we use in the laboratory is fluorescent in situ hybridization techniques. So in this, what we do is we take specific probes. It could be RNA specific for say Staph aureus or other bacteria which we want to detect. And this is one of the tissue from the infected patient as such. And Staph aureus, we have labeled it with fish, which is green in color. So wherever the arrow is, you can see that these are the biofilm being formed. Similarly in here, stained with different stains. These are some of the examples which of fish which we use in the laboratory. The other molecular techniques which we use is PCR and 16S RNA sequencing to further identify the biofilm forming bacteria in detail as such. The other common example of staining we use is the fluorescent staining. So this is again a stainless steel 316 SS which was sterilized, and this coupon was incubated with the bacteria which is Zen41. This is a Pseudomonas aeruginosa, and SAP231, which is a Staph aureus bacterium. So when these coupons was when we added the bacteria to these coupons and incubated for 3 days, after 3 days we removed the coupon and washed it with sterile PBS, and under fluorescent microscopy, when we stained it with the Cyto9, that is a green dye, we observed that the biofilm formation is there. And if you can see that wherever the arrows are there, specifically these kind of crevices, there is more of the biofilm formation. Even here, you can see that there's more of biofilm formation on these surfaces. So it shows that the surface roughness has more biofilm attachment as compared to the smooth surface as such. This is a review article which will tell you about all the methods what are being used in the orthopedic infections. Second is about the attachment on different surfaces, the orthopedic material as such. So we did the experiment in which we added the PBS, and to this we added synovial fluid. So synovial fluid basically is the joint fluid, which is in between the two bone joints as such. And what we did was we actually used the bovine synovial fluid, 10%. We added added it here, and we added the uh, Staph aureus, which is a GFP labeled Staph aureus. So this was stirred and placed on a magnetic stirrer, and with the help of the pump, the bacterial suspension was passed on to different coupons. So these coupons were made of different materials, and after different time intervals, we observed under microscope how is the bacterial attachment onto different coupons as such or different surfaces. So here you'll appreciate that I took hydroxyapatite, titanium, stainless steel, as well as polyethylene 4 surfaces. And the top row is with synovial fluid, the bacterial attachment at 5 minutes, 10 minutes, 15 minutes, whereas the bottom one is without synovial fluid. So you can see that the bacterial attachment is, or the biofilm formation is less as compared to the one in synovial fluid. So these are the biofilms which you can see, and these were quantified. For example, if you look at the polyethylene surfaces, so 5 minutes, you can see that the biofilm is very huge. So basically there's a chunk of biofilm which is attached, the cells attached, and they form a biofilm as such, or the aggregates as such. So by 15 minutes, the bacterial attachment takes place, and over time, you can see that there is these bacterial aggregates are very much as compared to that of without synovial fluid. So this suggests that the surfaces such as polyethylene are more prone to bacterial attachment and aggregation as compared to the other surfaces. So further we did the SCM analysis on titanium, stainless steel, hydroxyapatite, and polyethylene. So in presence of the synovial fluid, you can see that there is formation of the biofilm or the attachment of the bacterial cells, and so once the bacteria attach, the other bacteria which is coming, it can attach onto the surface, and there is interaction, cell to cell interaction with the host component that is present in the synovial fluid. Here you can appreciate that there is more of the bacterial attachment or aggregate formation as compared to the other surfaces. This is because the PE, it has more roughness as compared to the other surfaces. Next, coming to the bacterial aggregation. So the Staph aureus aggregation was observed in synovial fluid. In this experiment, what was done was we took saline, growth medium, and 10% bovine synovial fluid, and to this added the Staph aureus of 10 to 6 CFUs per ml. Yeah, and after 24 hours, we observed that there is no growth in saline, whereas there was growth but no aggregation of the Staph aureus. In 10% synovial fluid, you can see the chunk of bacterial cells. So these are bacterial aggregates which were formed. And we were interested to know what happens, what is it in the bovine synovial fluid, which helps in the formation of these aggregates. So further, we looked at different time intervals, at what is the time at which we can get these aggregates as such, and how fast the aggregation takes place and establishment of infection could take place. So this is the aggregated population of Staph aureus, which is attaching onto or forming the aggregates as such with respect to time. So without synovial fluid, there's hardly any aggregation, whereas at 5:15 and till 1 hour, you can see there is almost 60% of aggregation takes place. We also did the flow cytometry analysis, and based on the flow cytometry analysis, we observed that 1 hour is sufficient to have these aggregate formation. Even in the laboratory doing the microscopic experiments, it was observed that the Staph aureus aggregates in synovial fluid is much more higher within 1 hour as such. So 1 hour we thought is the optimum time at which the aggregate could form. Then we did the flow cytometry analysis, and it was observed that Staph aureus without synovial fluid, after 1 hour, we did the flow cytometry, and it was observed that there's hardly any cells which were forming aggregates as such. So whatever is there, you can see these small dots, these are just the planktonic cells, whereas this particular population, these are the aggregated cells, which is Staph aureus in presence of 10% of synovial fluid. So the other experiment what we did was to look for what are the different components which are present in the synovial fluid, which can help in the aggregate formation. So what we did was we used PBS, the whole synovial fluid, that is 10% of synovial fluid, fibronectin, fibrinogen, bovine synovial BSA, extracellular DNA, and hyaluronic acid. And using these components, we added the Staph aureus and looked for, after 1 hour, did the flow cytometry analysis and looked for aggregation pattern and observed that the fibronectin was the one which has the maximum aggregation, similar to that of the synovial fluid. Second was fibrinogen, whereas eDNA and hyaluronic acid had no effect on Staph aureus aggregate formation as such. So the host component, fibronectin and fibrinogen, was responsible for the aggregate formation. So next, coming to the intracellular Staph aureus formation in the host cells, that is the osteoblast. So as I said you earlier, that the bacteria can go inside the host cells and it can grow intracellularly. So similar examples were observed, wherein these are the fluorescent images of the biofilm, which is formed inside the osteoblast, which were inoculated, the osteoblast along with the bacterial cells, Staph aureus, and this is a GFP labeled Staph aureus. So you can see that dense biofilms were formed. This is a transmission image, and this is a composite image of both. So in order to eradicate these intracellular bacteria, normally vancomycin is being used at 10x MIC. But then even at 10x MIC, yes, you can observe that the fluorescent images shows green intracellular bacteria. They are still at a very high rate as such, and they're not being killed. So this is because the penetration of vancomycin is very minimal, and these antibiotics cannot go inside the osteoblast, and the bacteria remains protected. So we looked at in the laboratory different components, for example, piperine and CCCP. These are chemical agents as such, which can be used for increasing the permeability. So what we did was we added vancomycin at 10x MIC along with the piperine at 1x and observed that there is slight reduction in the intracellular bacteria, whereas vancomycin, when it was combined with CCP, there was a significant reduction in the intracellular bacteria as such. Further, coming to the bacterial count after the treatment. So with respect to the control, what was observed was gentamicin, CCP, piperine, and BMP2. So these are different components which different chemicals which we used. And yeah, so these are different chemicals which we used. And along with the vancomycin plus CCP, vancomycin plus piperine, and different combinations as such, dicloxacillin plus CCP, you can observe that there is significant reduction in the amount of intracellular bacteria which was present. So you can see that combination of the antibiotic along with the normal agent, that is piperine, or CCP, significantly reduces the intracellular bacteria. Then we looked at the different permeability of antibiotic in the osteoblast. Because as I told you earlier, vancomycin, it cannot go inside or it is difficult for vancomycin because it is a big molecule to penetrate inside the osteoblast. So this assay was carried out in which these are the osteoblasts as in control, and only the uh, different compounds like CCP, piperine, vancomycin, and combinations were added. And we added the red propodium iodide dye as such. So if the dye goes inside the osteoblast, it means that there is more of penetration of the antibiotic. So wherever you see the red color, it means that the compounds are very effective in intracellular accumulation of the vancomycin or the other antibiotic as such. So with control, you can see that there is no intracellular accumulation as such. Vancomycin also, there is no intracellular accumulation of vancomycin, or this decloxacillin is hardly any. Whereas when vancomycin in combination with CCP, there is increase in the accumulation of propodium iodide, which suggests that the combination of vancomycin and CCP, so CCP, it increases the permeability of the vancomycin. Similarly, with decloxacillin and CCP, there is increase in the permeability of the osteoblast with this particular combination as such. So along with this image, the percent uptake was mentioned and it was measured. So you can see as compared to the control, there is significant uptake of the red dye, and almost 80% of the dye was uptaken with decloxacillin and CCP. So it suggests that this combination can be used for killing the intracellular Staph aureus. The fourth one is about the host and neutrophil interaction with bacteria or biofilms. So normally the planktonic bacteria comes and attaches to the neutrophils. And since neutrophils are the host defense mechanisms, the neutrophil, what they do is they ingest the bacteria. That's, in this case, it's Staph aureus. So they ingest and they form the phagosome, and they degranulate or kill the bacterial cells as such. But what happens in case of if it is bacterial aggregates? So to understand that, the experiment was done in which the bacteria was incubated with soluble fluid, and after 1 hour, the aggregates were taken and then mixed with neutrophils. So in this case, this is a Staph aureus, which is GFP labeled, and the interaction with neutrophils, you can see that these are the interactions of the red is neutrophils. So the neutrophils are able to kill the planktonic bacteria, but the biofilms are still viable. So further to understand this mechanism, we did the flow cytometry analysis in which the Staph aureus aggregates and the planktonic bacteria were mixed with the neutrophils, and the percentage uptake was measured. So the neutrophils is 73.9%, and in case of the bacterial aggregates, when we when we looked for the aggregates and how much bacteria is present in these aggregates, so it was observed that only 26.1% were uptaken by the neutrophils, whereas if it is a planktonic bacteria, so almost 40.8% was uptaken by the neutrophils. So it shows that the bacterial aggregates, these are very less uptaken by the neutrophils, and they're protected because of the exopolysaccharide and the size of the biofilm as such. If it is a planktonic bacteria, they are very readily killed by these neutrophils. So far the outcome of the study I can summarize as the Staph aureus is often associated with orthopedic infection. Different molecular techniques such as FISH, PCR, 16S sequencing, these are the common techniques which are being used to identify the pathogen. Then fibronectin present in synovial fluid, it promotes the aggregate formation, and different compounds such as CCP and piperine It can decrease the intracellular Staph aureus and increase the permeability of osteoblasts to the antibiotics. The next one is Staph aureus aggregates. They can escape killing by neutrophils. So next, coming to the treatment of these orthopedic infections. So I was working as a consultant for biocomposites, and what this company they do is they supply these kind of kits, which are biometal loaded ones. So they have this calcium sulfate powder, which is a pure form of calcium sulfate, and antibiotics are basically added to the powder mixed together, and with this particular mat, different size beads are being prepared. So just to give an example, these are 4.8 antibiotic loaded biometal beads, and during the orthopedic infections, the surgeons what they do is they cut open the site of infection and they place these antibiotic beads at this particular infected site as such. So what happens is over a period of time, these antibiotic loaded beads will release the antibiotic and the infection will be cleared over time. So to understand what the role of these antibiotic loaded beads against different pathogens, biofilm forming pathogens as such, the experiment was set up for prevention and killing of biofilms. So we used 4 different bacterial strains, that is Klebsiella pneumoniae, which is carbapenem resistant, the another strain of Klebsiella pneumoniae. Then we used a strain which is a New Delhi beta-lactamase producing NDM1 positive ones, and Enterococcus faecalis vancomycin resistant biofilm forming strain. And we use different combination of antibiotic just to see that which combination will be effective in preventing or killing the biofilms of these test organisms. So the combination what we used was unloaded beads which didn't have any antibiotic, then rifampin at 600 mg per 10 cc in combination with vancomycin at 1 gram per 10 cc, then gentamicin and vancomycin and tobramycin, as well as rifampin and meropenem as such. So using these different combinations, what we did was we further formed the biofilms. So the first assay is biofilm prevention assay, in which we were interested to know the ability of the beads that was loaded with the antibiotic, whether it can prevent the biofilm formation after multiple bacterial challenges. So this is the petri dish, that is a 6 well plate was taken, and we added approximately 10 beads which had the antibiotic in the well, and the bacteria was added at 5 to 10 to 5 CFUs per ml. So since this is the prevention of biofilm formation, so the bacteria was added at the same time when we added the beads as such. So the complete media exchange was done after 24 hours. Fresh inoculum was added, that is depending upon the bacteria we used LB or another medium as such. So the CFU count was taken at day 1, 2, and 3. So these are the results of the residual biofilms which were formed at different time intervals. So these are different strains of Klebsiella pneumoniae, 1705, 2524, 2146, and this is Enterococcus faecalis. So this is challenge 1, that is day 1. We challenged it with the 10 to 5 bacteria. Challenge 2 is the next day. We removed the medium, again added the bacteria as such, and this is challenge 3. So this was to see how much effective the antibiotic beads are at different bacterial challenges, and whether they can prevent the biofilm establishment or formation as such. So log CFU counts, if you see that with respect to the unloaded bead, All the other antibiotic combination were very effective, even at challenge 3. Similarly, for the other, in case of the other strains, what we used, we can see that all the other combinations were effective in killing the or reducing the biofilm as such. Similarly, in this case, so initially it was very effective, but at challenge 3, so this is significant, R plus V, R plus M is significant, even V plus T is significant, but you can see that G plus V, that is gentamicin plus vancomycin, is not significant in this case. Similarly, for Enterococcus faecalis, all the antibiotic combinations were effective, and the most effective was R plus M. In most of the cases, it was R plus V and R plus M. So I'll come to that in the next. Okay, so this is the confocal imaging of the previous experiment, what we did. So at day 1 and day 3, you have all the 3 different strains of Klebsiella and Enterococcus faecalis. So the top one is unrolled beads. You can see that the dense biofilm were formed after 24 hours, whereas with all the combination, you can observe that the biofilm were completely eradicated at day 1. However, at day 3, you can observe that especially this combination, that is G plus V, there is still residual biofilm remaining. Even V plus T was not effective. Now, normally what happens is the surgeons, they use this combination, V plus T, a lot in during the surgeries. But we were interested to know, are there any other combination of antibiotics, for example, R plus M, R plus V, or G plus V, which can be used as an alternative to V plus T. So in this case, we can see that R plus M was much more effective in reducing the biofilm, or this is a prevention assay. So in preventing the biofilm, the combination of R plus M was very much effective. And just to say that everything in green is the live biofilm, and the other ones in red, if you see anywhere the red bacterial cells, so these are the killed or the dead bacterial cells, or dead biofilm as such. So coming next to the biofilm killing assay. So in the killing assay, what we are doing is we are forming the biofilm for 3 days. So we take the 6-well plate, add the bacterial culture to that along with the growth medium. 3-day biofilm is formed, and after the biofilm is being formed, we add the 10 beads, which has the antibiotic as such. So 10 beads per well is being added, and complete media exchange is done after 24 hours. And CFA count is taken at day 1 and day 3 after the addition of the beads. So 3-day biofilm plus day 1 and day 3. So these are the CFA counts of the residual biofilm of 3 different strains and of Klebsiella pneumoniae and 1 strain of E. faecalis. So you will appreciate that at day 1 and day 3, there's significant reduction in the biofilm, and you can observe that R plus V and R plus M, even here R plus V and R plus M, that is significantly reduces the biofilm to greater extent as compared to the other combinations, except just in this case, you can see that V plus T was more significant as compared to the other combination. So this suggests that using different combination of antibiotic, for example, R plus V and R plus M, we can significantly reduce the biofilm formation as well as the pre-established biofilms. So these are similar confocal images which we did of the 3-day biofilm which was formed. So the 3-day biofilm of this particular strains, and when we added the antibiotic loaded beads, so after that day 1, we observed the residual biofilm, and it was observed that R plus V significantly reduces the biofilm. Even R plus M was significantly effective in killing the biofilm as such. You will appreciate at day 3, where R plus V, there is no biofilm remaining. Almost 90% of the biofilm is gone. Even in case of R plus M, there is a complete reduction of the biofilm as such, as compared to the other combinations. So this is all the biofilm in green, and all the red is the dead biofilm. So based on these results, we suggested that yes, we can move from the original combination of vancomycin plus tobramycin. We can go to another combinations. We can use R plus V or R plus M. So in conclusion, this is the biofilms of antibiotic resistant strains of Klebsiella pneumoniae and E. faecalis were significantly prevented and killed by combination of rifampin plus meropenem, as well as rifampin plus vancomycin. And this was evident from the confocal images, which showed that biofilm prevention and killing substantially takes place. So in summary, I've shown that biofilm play an important role in the establishment of an infection. Culturing as well as advanced molecular techniques such as FISH, PCR, 16S sequencing are the important tools for identifying pathogens associated with the biofilm infections. And host components such as synovial fluid components, they promote the bacterial aggregation and biofilm formation as such. Again, the Staph aureus aggregates which were formed, they can escape killing by neutrophils. And in the treatment of the biofilms, I've shown you that compounds such as ACP and piperine, they can reduce the intracellular bacteria and increase the permeability of the osteoblast to the antibiotic, as well as the antibiotic loaded biomaterials could be beneficial in eradication of the biofilms in the orthopedic infection. So I would like to acknowledge Nationwide Children's Hospital and other people associated with at different places such as the Ohio State University, McGill, Pune University, University of Maryland, and the Department of Immunology and Microbiology at Colorado, and some of the funding agencies such as Biocomposites, OTA, and NIH, as well as NSERC back in Canada. If you have any questions, yeah, you can ask me. Thank you.
Thank you, Dr. Dusne, for your interesting talk on biofilms in clinical settings. The session is open for any questions from the attendees, if you have any questions. So, sir, there are a couple of questions from the attendees.
Sindhu, can I just ask a question to Dr. Devendra directly, please? If you're
Yeah, definitely, sir. Please continue.
Doc, I've enjoyed your talk. I just wanted a small suggestion whether how much the biofilms interfere with the bed sore healing. Because we do
Sorry,
have...
I didn't get your, I didn't get your last part.
The bed sores, sir, the pressure ulcers which formed in bedridden patients. So that we have a lot of problems treating the bed sores, especially in the elderly patients or patients who are comatose and bedridden for a long time. The pressure ulcers which form, we do have a lot of. We just use some antibiotics, and one of the good treatment which I have noticed as a dermatologist is the use of hydrogen peroxide, which does a little bit of oxidation. But how much is there? Are there any studies where biofilms are responsible for the non-healing of these ulcers, even the aphthous ulcers also?
Yes, there are a couple of examples, and you can find it online as well, that there are biofilms are basically everywhere, and based on the exopolysaccharide which they produce.
Yes.
So that's basically responsible for maybe neutralizing all the components as such of the antibiotics. So that is the first mechanism by which it stops the effect of the antibiotic. And as you said, that hydrogen peroxide, it is a very good example of treatment of the biofilms. What we have done is we had worked on one of the wound dressing, and that dressing is basically powered with a 6-volt battery. And we tried that on different, different animal models. So on dog, on pig skin. So it was infected with Staph aureus and Pseudomonas aeruginosa. So it was kind of a wound bed, which was there for almost 30 days or so. And after that, we added the wound dressing. It was basically silver-based wound dressing, and we powered it with 6-volt battery. And we observed that what this dressing does is it produces compounds which are similar to that of hydrogen peroxide, and it kills the biofilm as such, and even promotes healing of the,
Ulcers.
the cells as such.
So removal of the EPS is the basic thing of when you have to treat these. Thank you
Yes.
so much, sir. Thank you so much.
So we have one more question. So are there any commercially available biofilm inhibitors used in the treatment?
So there are different companies which are working in this aspect. So looking at different implant materials, as I said, that there are different materials which are prone to attachment. So right now there are different companies which are working on different materials as such. So it could be a combination of titanium as well as other alloys. So that is one aspect. Second aspect is there are companies which are working on different enzymes like dispersin. So one of the companies working on that aspect using dispersin enzyme which can disperse the biofilm EPS, and that promotes the effect of the antibiotic. Then the other example what I gave you about is uh, the one what I just shared with you about biocomposites. So these different beads which they use of 4.8 or different even smaller size, they can go into the crevices wherever there is biofilm is kind of it is not what do you say it's difficult to reach. So what happens is they can go to that particular places and there is sustained release of antibiotic. So whenever there is the upper layer of the biofilm is being killed, we have another antibiotic say dose as such, which is enough for almost. So we did some experiments in the laboratory, and these antibiotic beads they can retain the antibiotic almost for 30 days.
I have a question, Dr. Dusne.
Yes.
When you said about how is it, when you said about sustained release, what is your take on making nanoparticles which will have a slow and sustained release for a very long time? Has anything been done in that particular area?
Yeah, that's a good question. So different people are coming with different technologies nowadays, and nanoparticle is something which can be used in this aspect. So I remember that we were working on one aspect which was making nanoparticles from natural sources and utilizing those for sustained release. But then again, what happens is these nanoparticles, if you're putting it into the human body, they should be safe. So whatever biomaterials are being used, whatever biomaterials are being used by biocomposites. So what they're made of basically of calcium sulfate, which is a part of the bone as such. So when it degrades, it goes and it can be a part of the human system, or there's no negative effect on the human body as such. So those are a few things which needs to be tested as when you're talking about the nanoparticles.
The other question I have is, you showed about the biomaterial and the antibiotic, the company which you are collaborating with. What happens to the biomaterial? Is the biomaterial synthetic one or a natural one, or in which you have embedded the antibiotics?
Yeah, so the source is not clear to me, but basically this is a calcium sulfate. So as I said, this is a part of the bone as such, or it is similar to that of the body. So there's no negative effect on the host. And normally during surgery, what the surgeons they do, once they cut open the site of infection and they put these beads or replace the orthopedic materials as such, implants, and put these beads, they close it and then they leave it there. So they don't have to do again second surgery to remove these kind of biomaterials. And over time, these biomaterials become a part of the body as such. So they degrade and then it can be integrated in the bone.
One such thing which I read long time ago was hydroxyapatite, which they use.
Yes. But the problem with hydroxyapatite is it needs to be removed from the body. Since it doesn't degrade, so the surgeon has to again bring the patient into the OR, cut open that site, and then remove the hydroxyapatite. So that's again a second surgery or revision surgery. So that is not something which is not advisable.
Thank you, sir.
Yeah, thank you.
So we have one more question. Are there
Sure.
any infections from biofilms in rheumatoid arthritis patients?
Yes, that's a good question. So in rheumatoid arthritis, it's observed that there is more of eDNA, what you call it as um, yeah, it's observed that there's more of eDNA. So basically, it could be from bacterial or human source, and even we have observed that in these kind of patients, there is more chances of biofilm infection. So we are still working on that, and it could be that there is, yeah, depending upon the content of fibrinogen, fibronectin, and as I showed you, all those other ones. So to some extent, there is a role of eDNA also in biofilm formation. Surprisingly, it was not evident in our studies because maybe the content of fibronectin and fibrinogen was more in the synovial fluid. But if you take the patients with rheumatoid arthritis and look for biofilm formation in those kind of synovial fluid, yeah, maybe it will be a good option to look for the components.
So just thank you very much. Just one last question,
Sure.
sir. If it happens to see any biofilm formation in case of immunocompromised patients, so what kind of approaches, what different kind of approaches can be used in such kind of patients?
Yeah, so looking at the immunocompromised patients, as I said that there are a role of different neutrophils and other immune mechanisms for killing the biofilms. So in this these kind of patients, we have to look at the complete history, and normally the surgeons what they do is they look at the patient's history, and then they decide on the treatment mechanisms. For example, which are the antibiotics need to be given, what is the dose, and as you said, immunocompromised. So the dose will be much more higher as compared to that of the normal patients which are non-immunocompromised as such. So even there is different dose of the antibiotic loaded beads which are normally given in these kind of patients.
Sir, and this regarding non-steroidal anti-inflammatory drugs, can they be used for the treatment purpose?
Sorry, can you repeat the question again?
The non-steroidal anti-inflammatory drugs, can they be used for the treatment purpose to treat the biofilm?
They can be used. Uh, Regarding that particular treatment, I'm not sure about, so I cannot comment on whether it can be used or no.
Was a question
Again,
raised by some of the
yeah.
attendees. So
Okay, right. Because that is something which needs to be looked into. I'm sure there are different other non-steroidal components or compounds which can be used, but on top of my head right now, I'm not sure about.
and one more question, sir. Are there any home remedies that can be followed to prevent biofilm formation inside body?
Home remedies, there are, yeah, there are lots mentioned in Ayurveda. Just to give an example, there is honey,
Okay.
which is a good example, which even components from honey, and as I said, cranberry juice, which has CPAC and those kind of components. So whatever is being mentioned in the literature earlier, that okay, you use this one for curing the urinary tract infection or some other infections, but then there's no scientific evidence. So these kind of things are being studied a lot, and we have shown that yes, there are some components present in these kind of maybe Ayurveda or the other examples are like honey and cranberry juice, which can be used for biofilm infections as such.
I add one line in continuation of what Dr. Devendra said. In fact,
Sure,
I have a personal experience with regard to use of honey because very large sebaceous cyst infection at the back. And one of the leading surgeons from Mysore operated it upon, and it was almost it was about 3 to 3.5 inches length was the wound open, because it has to heal from the base, the whole thing. He used to apply for dressing only honey, pure honey we got, and he is a well-known surgeon, and he used only this as a dressing one, so that there is no further infection, and the healing process is, she was around 72, 73 at this age it was done, and I have seen a wonderful effect of it, honey being used more like a fantastic dressing material to see that no infection took place. She was not diabetic, but still everything went on very well, and that was actually a great revelation for me.
And one more question, sir. What are the underlying reasons for the difference in extent of biofilm formation in different materials?
So as I said, different materials, they have different surface characteristics. So it could be the surface roughness is one. So just to give an example, as I said, in P, that is polyethylene, there is more of a surface roughness, and because of that, there is attachment of bacteria, and biofilm formation takes place. Even charge is one which component which can be, so the surface charge is another one. Then there are some surfaces which are like copper, which are known to be antimicrobial. So these kind of surfaces can repel the bacterial attachment.
And one more question, sir. Are there any
Yes.
evidences of escape pathogens which are formed in the biofilms and resided inside the bones?
That's a classic example of osteomyelitis, in which what happens is the Staph aureus, it goes inside the bone, and they multiply there. So we have done some studies in which even after addition of antibiotic, the biofilm will be killed, but some of the cells inside the biofilm, they will change their genetic what do you call it? There'll be more of persister cells. So either they will form something like the spores. Just to give an example of that, so there will be more of going into a different growth phase, and the antibiotic will not be able to kill them. And when they find a suitable growth environment, they will again flourish and form biofilms. So that is something which we are interested in, like the persister cells within the biofilm.
So what is the difference between biofilm and biofouling?
It's basically just the concept. If you're speaking about biofilm in general, it is more of a medical as well as it's more of a medical term, as well as biofouling. If you're looking at that, it's more of an environmental aspect. For example, if you're looking at ship, so on the ship surface, which comes in contact with the water, so that is more prone to different initial primary colonizers, what you say about. So this could be bacteria, algae, and those kind of organisms. Then secondary biofouling takes place, in which there are like Pernaviridis and those kind of higher organisms. They go and they attach to the surface. And so biofilm basically, it can be used more of a medical term, and biofouling will be more in environmental aspect.
Okay, thank you
So,
very much.
thank you, sir. There are lots of questions, but we'll take only one last question before we move on to the
Sure.
next speaker. So the last question which I'm going to ask is, which is from a participant, if the rough surfaces favors biofilm formation, why is that there is biofilm infection in contact lenses?
Yes, so that's a good question. Usually what happens is, apart from the surface characteristics, it's also in which medium it is present. For example, in the eye, you have enzyme which is called lysozyme, and normally that is the one which normally kills the bacteria or the biofilm as such. In some of the patients or some of the people who are wearing contact lenses, over a repeated period of time, maybe that lysozyme concentration will be less, or wherever they're keeping the lens, that surface of the lens could become more prone to uh, attachment of, of the bacteria. Yeah.
Thank you very much, sir. Thank you very much for this.
Yeah, thank you.
Appreciate all the participants who have actively taken part in this, and there are a lot of questions. At some point, we'll post these questions to you, so you can probably answer them.
Yeah, sure. I'll be happy to do that.
Thank you so much.
Thank
What do
you.
you say?
I thank you very much, sir, for your interesting talk on biofilms in clinical settings. And now we move on to the next speaker. Over to Jamuna, please.
Introduce the.
Thank you, Dr. Devendra. I've always been admiring your work since my PhD days, and today I got this opportunity to have you here as one of our speakers. Thank you so much for addressing our delegates.
Yeah, thank you. Thank you for the invitation also.
Now we move on to our second speaker, Dr. Vandan Nagar. Dr. Vandan Nagar is currently working on as scientific officer in Food Technology Division in Baba Atomic Research Center, Mumbai. Dr. Vandan has been an high achiever since his student days. He was awarded gold medal for securing IS marks by the Gujarat University during his masters. Dr. Vandan pursued his PhD from Obi Baba National Institute, Mumbai, and completed his orientation course in nuclear sciences and engineering from BARC. Dr. Vandan qualified CSIR net, JRF net by CSIR, and also he has cleared the entrance exam conducted by All India test for MSc Biotechnology. He has been awarded international travel grants from DST India to present his research findings abroad. He has been awarded the prestigious Korean Atomic Energy Postdoctoral Fellowship to work at Kairi, South Korea. Dr. Vandan has 18 significant impact research papers, and he has also published book chapters in international peer-reviewed publications. And after joining Food Technology Division, BARC, Dr. Vandan has been working extensively on microbial safety as well as shelf life extension of food products using gamma radiation. His research work is focused on understanding molecular mechanisms involved in quorum sensing and biofilms by foodborne pathogens, and to develop various control strategies. This is a very brief introduction which I gave you about Dr. Vandan Nagar. His accomplishments are many, and he is very and highly inspiring to young researchers. Now I request Dr. Vandan Nagar to present his talk.
Good morning, everybody. Am I audible?
Yes, sir, you're audible.
First of all, I would like to thank the organizers for giving me an opportunity to present my work in this webinar. And after joining BRC, I joined the Food Technology Division in the Food Microbiology section. And over there, my objective was to study the presence of my objective was to study the prevalence of different foodborne pathogens in different food products and to devise the technique to control those foodborne pathogens in our food commodities. So with that background, I started my research work in Food Technology Division. So I will present my experience of working with Aromonas So my slide is visible. Over there, my first major aspect was to study the prevalence of the emerging foodborne pathogen Aeromonas and to study the methods by which we can control this foodborne pathogen. So the title of my today's presentation is Understanding Stress Response of Aeromonas in Foodborne in Food Environment and the various methods by which we can control this foodborne pathogens. So as we can see that Aeromonas is an emerging human pathogen which is related with causing gastroenteritis due to consumption of raw or undercooked salads, vegetables, fish, shrimp, and different types of food products and water. It is also responsible for causing extraintestinal infections like septicemia, wound infection, and cellulitis in different patients. Foodborne outbreaks related to this Aeromonas have been reported from various countries, and among children, Aeromonas is reported as one of the top 5 enteropathogens, which leads to diarrhea in children, and it leads to various deaths globally due to the consumption of this Aeromonas contaminated food products. Recently, Aeromonas has been reported as an emerging human pathogen. However, it is a very well established fish pathogen, which causes hemorrhagic septicemia and furunculosis in fish, and it leads to a significant financial loss to the fish industry, in which Aeromonas hydrophila and Aeromonas salmonicida are considered as the major fish pathogens. So with this background, I started my work, and in my initial days, I isolated this Aeromonas from different food sources, from fish samples, from river, and so we found that the majority of the information regarding this Aeromonas among in the literature is mainly limited to Aeromonas hydrophila, Aeromonas dhakaensis, Aeromonas sobria, and Aeromonas salmonicida. But there was very limited information related to the quorum sensing mechanism and the biofilm form, which is found by the Aeromonas, which is found in the food. So with this background and during my PhD work, I had isolated around 25 Aeromonas strains belonging to different species, and I wanted to know what is their biofilm forming ability and what are the different types of quorum sensing molecules produced by these organisms. So, so over the next 4 or 5 years, I focused on this work, and in which I wanted to study the stress response of Aeromonas in the food conditions. So I wanted to know what are the different types of quorum sensing molecules produced by this organism, what are the different types of biofilm formed by this Aeromonas food strains, what are the impact of different food related stress on this their biofilm forming ability. And since we are working on in a DAE, so our objective was how we can use this gamma radiation to kill this biofilm in different food industry. So I wanted to determine the radiation sensitivity of this organism in the biofilm condition. Then what are the different expression of virulence and stress responsive genes in this organism, and and in a specific study I wanted to study how this Aeromonas salmonicida strains behave, or they what is the reason for their means the type of Aeromonas salmonicida which I found, what is the significant difference with the already reported strains. So in Aeromonas, if we look generally, this quorum sensing plays a very significant role in the maturation of biofilm. So what is a quorum sensing? Quorum sensing is a population density dependent cell-to-cell signaling mechanism in which the bacteria recognizes the population density fluctuation, various changes, and depending on that population density, it regulates its gene expression. So in Aeromonas, it is already reported that it has a Lux RI type of quorum sensing system in which AHY is the autoinducer synthase and AHYR is the AHL dependent transcription regulator. In this AHL, it has a homoserine electron which is quite conserved, and it has a R group which can be of different carbon chain from C4 to C18, and it can also be there will be different type of substitutions like oxo and hydroxy substitutions are generally observed in the R group. So in all the organisms, means in the organisms which are based on Lux RI type of quorum sensing system, in the exponential phase, in the initial when in the exponential phase, HYI produces the HYI protein, which is responsible for synthesis of AHS from SAM and acyl carrier protein, and then this AHL binds to the HYR protein, and it also positively regulates its own synthesis. Where so in the when the cells are in the exponential phase, the bacteria will start accumulating more and more of AHLs, and when the concentration of AHL increases within the cells, then it starts secreting out the AHLs in the nearby environment, and during the early stationary phase, this AHL gets on accumulating in the environment. When the other Aeromonas or other Aeromonas will accept this AHLs, then this AHL will bind to the AHYR produced by that particular Aeromonas, and then it will regulate the various AHYR regulated genes in that organism, and based on that, the expression of various virulence factors, various enzymes, biofilm maturation will be regulated. In case of Aeromonas, quorum sensing regulates extracellular enzymes and hemolysin production, biofilm maturation, and type 6 secretion system. So to study the type of AHL produced by my different 22 Aeromonas food strains, I had to use AHL biosensor. These biosensors are of such type that they themselves don't produce any AHLs, but they can detect and they can respond to the AHL which is supplied in the external environment, and then they can show some phenotype which will be an indicator of the AHL which is sensed by them. So this is the structure of the normal AHL biosensor, in which the AHL will bind to the LuxR type of protein, and this AHL plus LuxR will bind to the promoter region, and this will give some change in the phenotype. So I have 3 different biosensor strains. One is Promobacterium violaceum CV026, one is E. coli PJA 130, and third is Agrobacterium tumefaciens, which will show different types of phenotype. Like Promobacterium violaceum will give the violacin production, E. coli will give me this fluorescence under the UV light, and one is Agrobacterium will show a different type of phenotype. So to detect the production of AHL by these different food strains, I cross-tricked my Aeromonas strains against this biosensor strain, and after 24 hours, they show some change in the phenotype. So here we can say that due to this this strain, 2654, I can see change in the pigmentation on the Aeromonas Promobacterium violacem. This organism is basically it doesn't produce any pigment, but when it detects AHL, then it will produce the violet pigment. In case of E. coli, we can see the light production, and in case of Agrobacterium tumefaciens, also it will show a change in color of the strain due to AHL detection. So I use 3 different biostrains so as to remove the chances of any false negative reports. And using these 3 different biostrains, I found that majority of my aromonas strains were able to produce AHLs, and thus it gives us the indication how whether they will be able to show different type of phenotype and whether they will be able to regulate this phenotype. So I further wanted to study what are the from this study I was able to know that my organisms are producing AHLs. So my next objective was to study what are the different types of AHLs produced by my this strains. So I first grew these organisms in MOPS buffer, and I extracted AHLs by a standard method in which we use ethyl acetate, and using rotavapor, I evaporated and finally got the AHLs, and the characterization of this AHL extract was done using C18 reverse phase TLC plate, and in this I use methanol water as the chromatogram. And based on the presence of and in this after TLC, I overlaid it with the Chromobacterium CV026. So wherever this here so here we can see that this is the TLC plate where I have spotted the extracts, and after the chromatography there was separation of these different types of AHLs, and then I overlaid it with CV026 biosensor strains. So wherever there was presence of AHLs, this CV026 will show purple pigmentation, and then we calculate the RF value of these different spots, and then in comparison with the standard compounds like C4 homoserine lactone, C6 homoserine lactone, we identified those spots. So based on my study, here we can see that I had different aromonas belonging to different species, and these are the different strains, and with different sources we found that among these different food strains, C4 and C6 were the major type of homoserine lactone produced by different aromonas, and but few this even numbered homoserine lactone are already reported and are found to be in the microbe, but this C5 and C1 were reported for the first time from our lab, in which we could identify this C5 and C7 homoserine lactones. So my aromonas strains were from different sources like sprouts, chicken, and the fish. Majority of my aromonas were observed from fish samples. So overall, we found that there was very high diverse pattern among the AHL production based on the source and species. Here we can see that from the same source also, like Aromonas KVA, A85 did not produce any type of AHL, but A91 produced 3 different types of AHL. So based on the species and a source, we did not find any specific correlation between whether it will produce a particular type of AHLs or it will produce a different type of AHLs. So my next objective was to, is there any correlation between the type of AHLs it produces and whether it is their biofilm forming ability? So biofilm is a complex community of viable and non-viable microorganisms which are enclosed by hydrated EPS, and these are anchored to biotic or abiotic surfaces, and it can be formed by us one or more microbial species. In food system, we found that this biofilms are found mainly in the drinking water distribution system and also in the food processing plants. And these biofilms are a major source of cross contamination in the food processing plants, and it can pose a threat to the food industry and the public health if this contaminated food is consumed by the consumers. As Dr. Devendra has already explained, there are five different stages of biofilm formation, in which the microbe will first reversible adhere to the surface, then it will lead to irreversible attachment, then slowly the organism will form a microcolony, and then during the maturation, this quorum sensing will come and play an important role, and it will lead to biofilm maturation, and then ultimately a dispersal of the biofilm cells will take place, and in which it will go to the next surface, and again it will making a biofilm. In case of food industry, we can the biofilm affects almost every part of our industries. It can be in the distribution of water, in the paper manufacturing, in the oil recovery, in the food processing, and in the medical implants. Since I'm more related with the food industry, I can say that this biofilm affects the acts as a continuous source of cross contamination and cross processing contamination. It reduces the shelf life of the product, and it leads to increased disease transmission, which will ultimately affect the food spoilage, product rejection, economic losses, and the cause various type of foodborne diseases. It also reduces the heat transfer and operating efficiency of the processing equipment. It leads to corrosion of metals in pipelines and tanks, and it sometimes it also leads to clogging of pipes in the food industry. So I wanted to quantify the biofilm which is produced by my aromatase food strains. So for quantifying this biofilm formation, there are different methods which are available. But since I wanted to compare different organisms across different stress conditions of pH, temperature, then the food preservatives, so I found that specific biofilm formation index is the most appropriate for my condition, in which I will take the change in the ODA of 570. This OD 570 is calculated by this crystal violet staining, and I will normalize this biofilm accumulation with respect to growth. And recently this specific biofilm formation index is widely used for comparing the biofilm formation across different conditions. And this SBF can be used for showing the degree of biofilm formation, which we can categorize the biofilm formers in no biofilm formers, weak biofilm formers, moderate biofilm formers, and the strong biofilm formers. So I grew my aromatase food strains in two different media. One is TSB, which is a nutrient-rich media, and M9, which is a minimal media. And I wanted to study how the different media composition affect the biofilm formation. And I found that I also quantified the biofilm biomass using crystal violet staining in the microtiter plate, and the specific biofilm formation index was calculated. So here we can see there was highly variable biofilm formation among the biofilm among the aromatase strains belonging even to the same species. Here we can see it is more in the case of triptic soya broth, whereas where in the few strains you will find more biofilm formation in M9 minimal media. So there was variable results among their biofilm forming ability, and a majority of these food strains were weak to moderate biofilm former under different conditions. And here also we could not find any clear correlation between the source or species of Aeromonas and their biofilm forming abilities. Since biofilm is a good ecosystem for the exchange of different virulent genes, antibiotic resistance genes, and also integrons, so I studied the prevalence of different virulent genes among these pathogens, and found that these pathogens had very high prevalence of various virulent genes like enterotoxins, acyltransferase, serine proteases, and our study showed that these strains are potentially pathogenic and pose a health risk to consumers due to consumption of raw or undercooked food products. In a natural environment or in the food environment, a biofilm formation is a combination of several factors. It as organism forms a particular type of biofilm on a particular surface. It is determined by various factors, like it is affected by the pH of the environment, in the microenvironment, temperature, nutrient, and the flow within that system. Then the surface properties of the, even for the of the microbe, and also of the abiotic or the biotic surface. And if we look at the food industry, then the what are the different food components which are the present in that particular microenvironment, and also whether it is a static system or it is a continuous flow is there. Then various other parameters of the bacterial cell, like physicochemistry, the species or strain, then what are the different EPS components produced, and what are the different gene expression or quorum sensing playing a role. And all these different factors play an important role in the biofilm formation. So I wanted to study the effect of different food related stress conditions on the biofilm formation by my strains. So I out of 22, I selected only 5 strains for the for studying this effect. So for this, I found that I grew these organisms in 3 different temperatures: 10 degrees, 30 degree, and 37 degrees centigrade, and I allowed them to grow, and I found that the maximum biofilm was formed at 10 degrees centigrade. It may be due to the lower movement of the organisms on at a lower temperature, which may give it more time and ability to bind to the particular surface. And among different pH conditions, I found that at the pH 5, they formed the highest in terms of SBF. It showed the highest biofilm formation, as at this pH, the organism may be in a stress condition, and it may be producing more and more of EPS, which will lead to the higher biofilm formation. And in case of sodium chloride, we found that the biofilm formation reduced as the concentration of NaCl increased. As such, Aeromonas is sufficient, means is able to grow to at least around 4% of NaCl, and thus it was able to survive in this condition. And if and then I also checked the biofilm forming ability in different food preservatives, and we found that as the concentration of food preservatives increases, though the quantity of biomass reduces, but the growth also reduced. So in terms of SBF index, we found that the biofilm forming ability increased with the increase in concentration of food preservatives. So our present study gives a baseline data for the biofilm formation under different stress conditions. And this baseline data will help food processors and regulators to optimize prohibitive measures to prevent biofilm formation by aromas in different food conditions. These organisms are generally under different stress conditions in the food environment, and there are no reports exponential growth rate. There are very few reports. There are very few reports which shows the gene expression of any organism under stress condition or on in the stationary phase condition. These food pathogens are generally in the are more closely to the stationary phase condition during their growing condition. So I wanted to study the effect of different food stress related events on the expression of housekeeping genes like RPOD, then the how the general stress response genes like RPOS and USPA are affected, and I took aerolysine as a virulence genes, and I wanted to study the different gene expressions. So RPOD is a sigma factor which keeps essential genes and pathways operational. RPOS and USPA are the general stress response regulators which are expressed under different stress conditions. And aerolysine is one of the major virulence factors of Aeromonas, which shows both hemolytic and enterotoxic activity. So the different stress conditions which I wanted to study was nutrient replenishment, nutrient deprivation, acid shock, and the cold shock. So in the next slide, I will show the experimental setup in which organism was grown overnight, and then it was means it was aliquoted into 5 different 5 ml culture, and then it was centrifuged, and then it was replenished with the in case of reference it was replenished with the spent tryptic soya broth. For the nutrient replenishment, I replenished it with the fresh tryptic soya broth. For the nutrient deprivation, I replenished it with the saline. For acid shock, I gave spent TSB whose pH was adjusted to 4.5. And in case of cold shock, it was given spent TSB at 8 degrees, and then it was further allowed to grow. And RNA was also isolated. So we found that there was no significant reduction in the cell count of both Aeromonas strains. Here I compared Aeromonas hydrophila 839, which is a type culture, and my food strains Aeromonas hydrophila 8331. And we found no significant reduction in the cell count of both strains due to all this stress treatments. Then in case of after giving different stress conditions, RNA was isolated from this bacteria, and then the gene expression was quantified, in which 16S rRNA was kept as a reference gene, and relative gene expression was determined using 2⁻⁻ delta delta CT method, and the significant expression was considered if it is more than 1.5 log to the base ratio or less than 1.5 ratio. So here we can say, see in case of nutrient replenishment, significant upregulation of RPOD in both the strains was observed, and similarly USPA was also shown to give the significant upregulation. This shows that when we give the nutrient replenishment, the cells are actively growing into the cells, and in case of USPA, it shows that when organisms transit from a stationary to a newly replenishment condition, there is some transition in which USPA plays an important role. In case of nutrient deprivation, we saw an upregulation of RPOS and USPA genes, and these two genes are already known to play an important role in case of nutrient deprivation and in the stress conditions. In case of cold shock, there was significant induction of RPOS. It may be due to the stable structure of RPOS RNA in the cold condition, and which will give it a stable structure, and it can live maybe giving the stable RPOS. And in case of acid shock, we found a significant role of USPA. So overall, we found that there was a significant induction of various type of genes when the organism was given a different type of stress, and it shows that the organism is preparing when a organism comes is exposed to a particular type of stress, then it prepares itself to handle the next round of stress. During my research during my initial work, when I was doing isolation of the different Aeromonas strains, I found that majority of Aeromonas salmonicida strains which are reported in the literature, they are all are psychrophilic in nature, whereas what Aeromonas salmonicida I found, they were quite easily able to grow at 30 degree and 37 degrees centigrade. So in the literature, Aeromonas salmonicida is divided into 5 subspecies like Aeromonas salmonicida, Smithia, Mothocida, Acromogenes. These are mainly the cytophilic in nature, which are reported from the deceased fish from cold water. Whereas only one subspecies of Pactinolithica is reported, and it is known to be mesophilic in nature. But when I did the biochemical test, I found some difference in their motility and the pigment formation with respect to Aeromonas salmonicida subspecies Pactinolithica. So I collaborated with a research group in Canada, and we did a complete set of biochemical tests, and we found that my Aeromonas salmonicida strains were showing few variations as compared to Aeromonas salmonicida strains which are already reported and classified. So we did the whole genome sequencing of these 3 strains, and we also did the growth characteristics of these 3 strains at 37 degrees centigrade, 18 degrees centigrade, and 7 degrees centigrade. And we found that my 3 food strains Y577, Y567, and Y47, along with Aeromonas Pactinolithica, were able to grow efficiently as compared to other Aeromonas salmonicida strains at all the different temperatures. This is 37, which is mesophilic temperature, 18 degree, which is in range between mesophilic and cyclophilic, and at 7 degrees centigrade. So my mesophilic strains grew more efficiently than cyclophilic ones, even at all the 3 different temperatures. And so we sequenced these 3 strains and compared with the already sequenced Aeromonas salmonicida genomes, and we found that my Aeromonas salmonicida formed a different cluster with the mesophilic Aeromonas salmonicida subspecies. And on doing the whole genome analysis, we found that Aeromonas salmonacida are highly diverse with members from mesophilic, then these are the intermediate and the psychrophilic lifestyle. So the earlier literature showed that the Aeromonas salmonacida were able to grow at a low temperature. Here we can see that our isolates are more toward mesophilic, and the earlier reports were psychrophilic. So our study showed that Aeromonas salmonacida will not be found only in the psychrophilic conditions, but even in the mesophilic condition also, Aeromonas salmonacida can survive and grow. So in the food industry, these are the different methods which are used for controlling biofilms. So here we can, in industry, we can see different chemical treatment, enzymatic disruption, pre-coated materials are used, and biosurfactants, bacteriophages, bacteriocins, use of essential oils, then quorum sensing inhibitors. All these different methods are used for controlling biofilms. So since I'm working in a Department of Atomic Energy, my interest was to study the radiation sensitivity of Aeromonas in planktonic versus biofilm formation on different surfaces. So we designed a co-incubation apparatus in which we can see here in the Falcon tube, a broth is there. So it will give us a planktonic condition. Whereas here I have used a glass slide, or in the other case, I also used carrot, which will be used for as a substrate for surface for adherence of the Aeromonas cells. And then after allowing it to form biofilm, I gave the different doses of gamma radiations, and after giving this gamma radiation, I determine the D10 value. So D10 values is a indicator of the radiation sensitivity of this bacteria. So here we can see this radiation sensitivity is determined in the term of decimal reduction dose. So this D10 value is the dose which will kill 90% of the population, and it is here we can plot the log value of the number of surviving organisms, and here we can have the dose in gray. And based on this, we can find the D10 values, which is D by log n0 minus log n, and it will be measured by the slope. So D10 will be minus 1 by m of this slope. Means this slope will give us the m value, and based on that we will find the D10 value. So the higher the D10 value, it shows that the organism is more resistant to gamma radiation, and the lower decimal reduction dose will give D10 value, will show that this organism is sensitive in the this particular condition. So here I determined the D10 values of Aeromonas. So here I took two different strains, Aeromonas hydrophila A331 and Aeromonas salmonacida Y567. And in this, I determined the D10 value in broth, which is basically a planktonic condition. Then glass, which is the biofilm which is formed on the glass surface, and this green is the biofilm formation on the carrot. So by determining this D10 values in three different conditions, I observed that there was no significant difference in the D10 or the radiation sensitivity of both the strains A331 and Y567 in case of planktonic or on the biofilm or the biofilm formed on the glass surface, whereas there was significant difference in the radiation sensitivity in the planktonic as compared to biofilm formed on the carrot surface. Whereas for the other Aromonas salmonicida Y567, there was no such significant difference in case of in the planktonic and on the biofilm formed on the carrot surface. I also did the SEM analysis of the biofilm formed on the glass surface and also on the food surface. Here we can see on the food surface, on the carrot surface, we can see the biofilm formed by Aromonas hydrophila on the carrot surface. So based on our study, we can say that the radiation sensitivity of this Aromonas species is affected by the growth form, whether it is in the planktonic condition or in the biofilm condition. Then the nature of attachment surface, like it is adhering to the glass surface or to the any food surface, and the strain which we use for study. Like there is a difference in the behavior of Aromonas hydrophila and also for Aromonas salmonicida. So based on this study, I found that Aromonas food strain show high diversity in their AHL and biofilm formation under different conditions. However, low temperature and acidic conditions resulted in higher biofilm formation. Then the radiation sensitivity is affected by the growth form, nature of attachment surface, and the strain used. And the induction of different stress regulators during initial stress events may prepare a cell for surviving and sustaining any subsequent stress. And Aromonas salmonicida species is a highly diverse with members of mesophilic, intermediate, and cyclophilic lifestyles. So this work was done by me before going for the postdoctoral work. Then after returning back from Korea around 1 year back, I started work on bacteriophages. These bacteriophages are the viruses of bacteria which will kill the specific host, and by this way we can specifically kill the bacteria which we want to kill. So these bacteriophages are widely means nowadays bacteriophages are widely used for controlling the bacterial organisms. So this bacteriophages contains capsid, and it has a genetic material in its head. It can be DNA or RNA, but in case of bacteriophages, mostly DNA is the genetic material. And based on their morphology, we can classify into different families are there: Myoviridae, Podoviridae, Cycloviridae. So different classification we can make. So these bacteriophages are widely used for killing the for removing the biofilms, in which certain bacteriophages are also known to produce some enzymes, and these enzymes can be of depolymerase in nature, and they can be used to remove the EPS, and then these bacteriophages themselves will go and attack the bacteria, and thus it will help in the complete removal of the biofilm from the food system. There are certain products which are already approved in case of for the food application. Different 5 products are approved, and among these are mainly they are against E. coli, Listeria, Shigella, Salmonella. But none of Spage cocktail related to the Aromonas has been approved. So we are working. I have started working on the isolating phages which can be of broad range specificity, and which can be used for killing the Aromonas in food system. So I started isolating phages from different sources. Again, these are the different host cells of different species: Aeromonas dhacanensis, Hydrophila, Gende, Salmonicida. And this is the water from sewage treatment plant. Then one of I collaborated with one person, so they provided me water from Ganga, and from zone 1 and zone 2 also we found that this water and the sewage treatment plant water contained some bacteriophages which were effective against Aeromonas dhacanensis. And also they also had some phages which were effective against Salmonella and Gender. Whereas in river water, I found phages against Aeromonas salmonella and Aeromonas hydrophila. So these are the different phage characteristics which you can see against different host, and this phages needs to be purified, and then later on they can be characterized. So against Aeromonas hydrophila 839, I have around 7 phages, and against Aeromonas dhacanensis, I have around 12, 13 phages are there. And among this, then I also did the host range analysis of these phages against different host cells of different species and strains belonging to Aeromonas hydrophila, and also I tried against E. coli whether they are able to infect already known E. coli. But we found that these phages are very host specific, and they will show lysis against the host only when a particular host is when they are subjected to a particular host. Similarly, in case of Aeromonas dhacanensis phages, we found that they were also highly specific and were showing lysis or were showing lysis only against their respective host. So based on the phage killing assay, means out of those 7 phages against Aeromonas hydrophila and around 12 phages which were against Aeromonas dhacanensis, we performed phage killing assay, means which phage was able to effectively kill the host cells. So based on that, I have selected 6 phages, 2 against Aeromonas hydrophila, 1 against Aeromonas hydrophila 8331, which is my food isolate, 2 phages against Aeromonas dhacanensis, which is a clinical isolate, and 1 phage S1 against my Aeromonas salmonella food isolate. So I have decided to characterize these 6 phages, and for this we have already prepared DNA samples for the whole genome sequencing, and we have already given for the sequencing part. And out of this, for phage S1, we have already sequenced it and we have submitted to the NCBI. And due to lockdown, we are facing a lot of problem in TM analysis of our phages. But slowly we are, after opening of this different facilities, we are hopeful that we will be able to get the TM analysis. So once we get the TM and the whole genome sequencing, we will be able to know which phages are quite capable of being of useful for the food industry. And then based on that, we will be doing the temperature, pH, and different conditions which they are exposed to in different food industry. We will see whether they will be able to survive in those conditions, and based on that, we will make a cocktail of different phages so that they can be used in the food industry. So this was one of my work which I have started related to use of bacteriophages in controlling biofilm in the food industry. Along with this, I have also started some basic work which will be related to inhibiting the various quorum sensing system in aromas. So if we can inhibit and design, or we can find some molecules which are able to inhibit the quorum sensing system, then it will be of useful in the different systems. Even in the medical system also we can use it. So for this thing, we started with the basic characterization. This is the in silico analysis of ADHI. ADHI is the aromatase dhakaensis homocysteine lactone synthase, and ADHR is the aromatase dhakaensis transcription regulator. So based on this, we found that this amino acids, these proteins are very important, and they are quite stable, and they are found in the cytoplasm, and we can purify them. So I started with cloning that ADHI gene in the PET21, and after cloning, I transformed it in BL21DE3, and here we can see that the normal BL21DE3 did not show any pigment formation when it was strict alongside the Chromobacterium CV026, which is a biosensor strain, whereas my three different clones, D13E, D13F, and D13G, they when they were strict along with the biosensor strain, so this gene was cloned and properly expressed in the transformed cell, which is showing the production of this violation pigment formation. So we are now we are optimizing the expression and purification of this ADHI in different cells, and we found that this fan band is showing the ADHI, but along with this we are also getting some other proteins. So now we are in the process of purifying this other protein, so that we can produce this ADHI in a large quantity, and then we can use it for some structure analysis, or we can determine its structure. So these are to the basic work which I have started around 1.5 years back, and right now they are under process. And in the end, I would like to acknowledge my section head, Dr. Shashidhar, and my lab assistant Lipika for their help during my research. Thank you. Any questions are there? Then I would like to answer that.
Thank you very much, sir. Thank you very much, Dr. Nandan. That was an exhaustive work, and you've done a lot of work on genomics of microbial. There are a couple of questions. In fact, there are lots of questions. We'll take up only a couple of them. Right. Would all types of biofilms be infective only because there are microbes present naturally in the body? Would all be pathogenic? Would all types of biofilms be infective only because there are microbes present naturally in the body? Would all of them be pathogenic?
No. All the microbes which are present in our body are not pathogen. So depending on the different virulence genes present in them, based on that only they will be able to adhere, and then they will be show any type of pathogenicity.
Okay. So there is another question. Have you designed any combinatorial treatment methods to combat the Aeromonas biofilm formation?
Yes, we in a long run we want to use a combination of bacteriophages along with the very low dose of gamma radiation, so that this combination treatment we can use for treatment in the food processing plants. So we have seen that gamma radiation will take care of the very low dose of the presence of very low number of bacteria, and for the specific type of pathogens we will use bacteriophages. So this combination will turn out to be a very effective one.
Okay. So will the food not get affected with high dosage of gamma?
No. We have already known that around 2 kilograms of dose doesn't affect the nutritional and the organoleptic properties of the food products. So even we want to reduce from 2 kilograms to around 500 gray. So for that we have to use some combination treatment. So what we are thinking that around 500 gray will take care of the normal microflora. But if the food contain any specific type of pathogen, then if we want to remove the pathogenic bacteria, then we have to remove those organisms completely from the food commodity. So for that removal, we have to increase the gamma radiation. So instead of increasing the gamma radiation, if we use the bacteriophages, then we can reduce the gamma radiation dose. So this combination will be much more effective.
And one of the question is, why was ethyl acetate used specifically for extraction?
This ethyl acetate is a a solvent which will extract this AHS. Means they are amphipathic molecules. So they have both aliphatic part also, and this hydrophobic and hydrophilic, both parts are there in the AHLs. So it will take care of them, and it will extract from the aqueous system. Since our media is in the water, means it is in the aqueous system. So from that, if we want to extract the AHLs, we have to use the solvent which will be able to mix with the aqueous solution, and then it should be able to provide the AHLs.
Another question is, the bacteriophage which we have used in several experiments, will it not affect the normal microflora which is present in the gut?
In the gut, mostly for the food system, we are targeting. Means, see, if you see my work, or if you go to the literature, you will find that these bacteriophages are very specific to their host. Let's say if a bacteriophage is, even if we screen and search for a lot of bacteriophages, so we can, at a maximum, we will find a bacteriophage which will be able to infect a particular organism. Let's say salmonella. So it will infect only salmonella. It will not affect E. coli or the other normal microflora which is present in our gut. So this specificity we can use. So this type of, which are this specific bacteriophages which we want to use against a particular type of pathogenic bacteria, then it will not affect the normal microflora of the human.
Thank you, sir. There are a couple of questions from Dr. Jamuna. Jamuna, over to you.
So there is a question here. Aeromonas can even reside in low oxygen concentration. Will the canning process contain this bacterium?
Pardon?
Aeromonas can even reside in low oxygen concentration. Will the canning process contain this bacterium?
Yeah, in the yeah, it can contain canning, but at what conditions we do it, that will be effect. Means we have to see it.
Okay. And there is one more question. In case of resistant pathogens, what quantity of radiation is fine with respect to food quality?
Means means if we speak, we have to optimize the food commodity and also the radiation treatment which we want to give. So based on that, we have already it is already in the FSSAI regulation, in which all the food category, all the food products in India are categorized into 8 different classes, and depending on the usage, means whether you want to increase the shelf life of the food product, or if you want to kill any specific type of pathogen, or any further any additional benefit you want to get from this, different dose ranges have been given. So for majority of the products, around 1 kilogram to 2 kilogram is quite sufficient to for the killing of the different type of pathogens. Like means in the mango season, if we export around 1200 metric tons of mangoes from India to USA. So over there also we use around 300 gray of dose is sufficient to sufficiently decontaminate the surface, and also for the killing the insect which are generally found in the mango seed. So means depending on the usage, we can have a different dose range we have to use.
There is one more question. Which media favors biofilm formation? Cryptic soya broth or minimal media? And why is there difference in biofilm formation? Is it genetic mechanism or environmental factors?
Biofilm formation is not a single gene event. So if we cannot say that if a single gene is present, then this organism will be a strong biofilm former or a weak biofilm former. So we have to see different conditions, and also depending on the surface on which the biofilm is formed, and also the genetic makeup of the organism and the microenvironment, means the different type of pH, temperature condition. Depending on that, this biofilm formation will be means the quantity of biofilm formation will be determined. And among this TSB and M9 minimal media, if we compare, generally more biofilm will be formed by in the tryptic soya broth, because it will be more nutritious. So it will give more nutrient and the carbon source for the organism to produce EPS. But if you give the same carbon source in the minimal media, let's say 0.4 to 0.8% of any carbon source in the form of sugar, if you give, then even in the minimal media also you can get a higher biofilm form.
Okay. Thank you, Dr. Vandan Nagar, for taking up the questions. And thank you for the insightful talk and enlightening us about the use of gamma radiation as well as the bacteriophages for controlling biofilms in food. Thank you so much.
Thank you.
Now I request Dr. Asha to introduce our next speaker, Dr. Veda Hegde.
Thank you, Dr. Jamuna. Moving on to our last speaker for today's session is Dr. Veda Hegde. Dr. Veda Hegde is currently professor and head of the Department of Oral and Maxillofacial Pathology and Oral Microbiology, SDM College of Dental Sciences and Hospital, which is a constituent of Sri Dharmasthala Manjunatheshwara University, Sattur, Dharwad. Dr. Veda Hegde got her BDS degree from JSS Dental College. So we have a proud alumni from this in this webinar series. And she completed her MDS from Government Dental College in Bangalore, securing 28th rank in Karnataka State Entrance Examination. She has obtained ICMR research grant for postgraduate dissertation in the year 2018. She's also a co-investigator in one of the research projects related to oral cancer. Dr. Hegde has guided several PhD students and also has published a book on apoptosis in oral dysplastic lesion and oral squamous cell carcinoma. She has won several awards for this kind of research. Dr. Hegde has published over 25 publications in national and international journals and has been a reviewer for reputed journals. And she's also a national coordinator for International Forensic Odontology and Child Abuse Conference in King George Dental College, Lucknow in the year 2019. She has been an external evaluator for research proposals for most of the government grants. And she is also a member of Board of Studies at Dharmasthala Manjunatheshwara University, and has conducted several continuous dental education programs, webinars, clinico-pathological meetings for undergraduate and postgraduate studies. And also recently got to know that she is a good athlete as well. So with this brief introduction, today's talk will be on whether the biofilm is a boon or a bane. Over to you, Dr. Hegde.
Thank you so much, madam, for this introduction. Am I audible to you, madam?
Yes, ma'am.
Yes, I'll share the screen now.
Sure.
Is it visible, madam?
Yes, ma'am, it is visible.
Yes. A very good afternoon to all of you here. And I thank the organizers for giving me this opportunity in this teaching learning process. And as madam said, I'm really proud because I'm an alumnus of this JSS Dental College, which I have studied as an undergraduate student. So without wasting much time further, I'm going to start with my presentation, that is oral biofilms, a boon or bane. Biofilms are ubiquitous. They are present literally everywhere, from the frozen deserts of Antarctica to the depths of the ocean, as well as deep within the surface of the earth, miles from the surface of the earth, to small areas like our oral cavity. They are present literally in all surfaces. Now, biofilms grow only on or are seen only on those surfaces where there is a surface where they can attach the microorganisms, a nutritive component that they are made available with, or a fluid environment where they are able to thrive, attach, and grow. It is seen that more than 50% of Earth's biomass is present in the form of biofilms, and it is seen that in more than 95% of these bacteria that is present in the biomass exists actually in this biofilms. Now, if you see at this photograph, you can see that the habitat is an aqueous habitat where the blue and the red represent cells that are colonizing or trying to colonize a surface of the rock. However, only the red ones are able to colonize, and the blue cells here are choosing or taking other areas to colonize because they're not able to grow or thrive in this particular area. The human body consists of more than 10 to the power 14 cells. It's quite surprising to know that only 10% represent the mammalian cells. That means to say that a majority of 90% don't represent our cells, but it actually consists of microorganisms residing in various locations in our system, that is skin, mouth, digestive tract, and the reproductive tract. And we need to know about them because they have broad implications in the life that we lead. And even they have profound influence on the general health of us. So it's utmost important to know that these biofilms have an impact on health as well as disease process. Now, the oral cavity is warm and moist, which allows a complex ecosystem consisting predominantly of bacteria to thrive in this particular area. Other than bacteria, which constitutes more than 170 species of them, we have viruses, fungi, protozoa, and mycoplasma also. These microorganisms colonize the soft tissues, that is the mucosal surfaces, as well as hard tissues, that is tooth surfaces, forming a structural, very well organized multi-species community called as oral biofilms. Now, oral biofilms are beneficial to the host, and when the microbiota of the biofilms change, there is issues that is related to oral diseases that can cause because of these biofilms, which are called as plaques. So it is noted that more than 85% of the dental services that we take it accounts to diagnosis, prevention, and treatment of oral biofilm related diseases and their sequences alone. Now, why do we require these microorganisms within our oral cavity? As we know that the organisms help us, as I told you, so there is something called as colonization resistance, wherein the microorganisms present within the oral cavity prevent exogenous organisms, mainly pathogens, from residing and causing a disease process. And this occurs by competing with them for nutrients or attaching to binding sites or receptors and making them unavailable for the organisms to attach, or releasing antagonist products like bacteriocin, hydrogen peroxide, which makes them an unfavorable microenvironment where pathogenic organisms cannot survive. They are also involved or implicated in the development of the host tissues. That is, they stimulate the host defense mechanism and also are involved in oral mucosal development. There is a constant communication happening between the oral mucosal organisms as well as the host tissue. To give an example, certain species of streptococci are known to inhibit epithelial cytokine synthesis, that is, they suppress it. And they are also involved in inflammatory epithelial cytokine synthesis, affect the cytoskeleton of the cells and cell-to-cell attachment. There is another component that is of use is the nitric oxide pathway. Now, in this system, what happens is 25% of the nitrates that is excrete or secreted into the saliva is converted into nitrite by facultative anaerobes residing within the oral cavity. Now, what happens is this reduction of nitrates to nitrite is going to be beneficial for us because modulation of the blood pressure and the blood flow is due to this process. Further, this nitrite can further get broken down into nitric oxide when they come in contact with the acidic pH of the stomach, following which it stimulates gastric mucus secretion, which is beneficial to us. And even they are known to have antimicrobial properties, that is, acting against microorganisms. So there is a plethora of host defense mechanism given by these residing organism against pathogenic species trying to invade our tiny oral cavity. Now, as already mentioned by Sir before, that there are many factors that are going to have an effect or going to be the deciding factor. What are the organisms going to reside in these biofilms and how stable resident they are able to form so that they can have beneficial effects on the host. It includes nutrition, temperature, pH, host defense mechanism, and the others. To quote one example, from a dental point of view, these biofilms that are seen in an otherwise normal state, there is inflammation happening if in case there is a small biofilm which is usually seen in close proximity to oral soft tissues residing on the tooth surface in close proximity to the soft tissue. What happens is inflammation is there, but we have protective factors within our saliva and the mucosal tissue that keeps the equilibrium properly so that we don't have any pathologies residing. However, if our oral hygiene is not kept properly, or if there is any pathology related to salivary glands which does not give us the benefits of saliva, then what is going to happen is there is a gradual shift in the microbiota of these biofilms into pathogenic forms, which are going to elicit an inflammatory response along with toxins and other pathology within the oral cavity. Now, this can range from the pathological process that sets in because of an alteration in the environment, can range from being immunological to non-immunological in nature. That is to say that in case if it is an immunological reason for a change in the environment, it could be that there is immune suppression for some reason, or there can be a neutrophil defect, which can alter the environment within the oral cavity, wherein there is a change in the organisms that are residing. Likewise, non-immunological methods or non-immunological reasons why there is a change in the environment could be, as I said, one thing that salivary glands can be having an issue, or they can be increased intake of carbohydrates, especially the carcinogenic substance, that is sucrose, where continuous uptake of this can cause, or poor oral hygiene managed by the particular person can change the microbiota in these biofilms in such a way that the community of organisms that are housing in these biofilms rearrange them to survive a different microenvironment and pose a risk or cause diseases within the hard tissues as well as the soft tissues. Now, it was Bill Costanton in 1978 who is actually called as the father of biofilms. He gave the biofilm concept. Basically, as you know, he is a microbiologist who actually loved traveling and moving up the hills and visiting natural habitats. So during one of his encounters, he saw that on the surface of rock where water was flowing, he saw a living film being present. And that's how the whole concept has been changing over time with new innovations and new methods of detection. So this biofilm is a dynamic environment and it is biologically active. So it consists of numerous organisms present within them that are attached onto the surface of the tooth with respect to the oral cavity that I'm talking, and function as a coordinated, organized microbial colony. Now here in this schematic representation also we can see that there is a tooth surface here and there is a big colony of microorganisms of varying types literally stuck to the surface of the tooth. Now the characteristics of biofilm is such that the organisms are able to take nutrients, they have water and all the requirements like enzymes and other nutritive component to grow as well as to ward off unwanted molecules that can harm them. For example, chlorhexidine is taken as an antimicrobial mouthwash by many of us. Now this antimicrobial agent may not act in the given concentration because the organisms have made an environment wherein they are able to restrict the entry of these antimicrobial agents. And these organisms live in synergism or an antagonistic interaction between various species of cells. For example, Streptococcus will use the carbohydrate component and give rise to lactate. And this lactate can be used by other organisms like Veillonella. Likewise, organisms that are anaerobes can still persist in an aerobic environment by its close association with organisms that take up oxygen or aerobes. So when the aerobes take up the oxygen in a given microenvironment, the anaerobes are able to thrive in the absence of oxygen. Likewise, certain organisms might synthesize or might have enzymes that are able to catch up this oxygen way that creating an anaerobic environment for the organisms which can thrive in this situation to flourish. Now, basically, the structure of the biofilm has already been told by sirs. I'll just do it a little faster for this. Now, they are existing in microcolonies and they constitute only 15 to 20% of the entire biofilm, whereas the matrix constitutes the majority component on which they are seen literally attaching or adhering to the matrix. Each colony behaves like an independent community and has
Hello, ma'am.
Microenvironment, there are water channels that this circulatory system is going to provide nutrition to the organisms to live, as well.
Ma'am, we are not able to hear you. Mute yourself.
Ma'am, can you please unmute?
Can I call him now?
Yeah, you're audible now.
Just 1 minute, ma'am. I'll share the screen. Are you able to see me, madam, my screen?
Your complete desktop is visible, ma'am.
Just 1 minute. Just 1 minute, ma'am. I'll just share it.
Sure, ma'am.
ಮೊನ್ನೆ ಶೇರ್ ಆಗ್ತಾ ಇಲ್ಲ ನೋಡು. ಆಗಿತ್ತು. ಇಲ್ಲಿದೆ. ಇಲ್ಲ ಅದೇ ತಾನೇ ಆಯ್ತು. ಮಾಡ್ದೆ ನಾನು ಕನೆಕ್ಷನ್ ಹೋಯ್ತು. Madam, are you able to see me now? The screen?
I'm able to see you.
Yes.
What I can do is until here. Okay. Yes, your slides are visible now.
Yes, madam. Thank you so much. Now, these microorganisms are the ones to synthesize this matrix as well as degrade whenever required. And it contains both organic and inorganic materials present. The organic material consists of proteins, glycoproteins, lipids, albumin, and many other components. And the inorganic components usually consist of calcium and phosphate. And in minor quantities, there is fluorides, potassium, and sodium. These organic and inorganic components are basically derived from the saliva, gingival crevicular fluid, and the bacterial products. For example, the polysaccharides that are present within the matrix is a derivative from the bacterial byproducts, while albumin comes from the gingival crevicular fluid. So in this way, and the glycoproteins are a derivative from the saliva. So one example I have given for each of these to tell that they can take the material from any of these sources that are components of the saliva. Here I have kept, there is a thin film of the biofilm that is there. Here it appears yellowish to gray in nature. But if you see the next photograph here, the oral cavity is relatively clean. It's very difficult to see here any discoloration that way, which indicates a presence of a biofilm. Now, this visibility of the biofilm can be enhanced by use of certain disclosing agents. Now, the disclosing agent used here is the erythrocin dye that is taken up by these biofilms. So you can see that in majority of these areas that the biofilms are forming, They are in close proximity to the soft tissue. This red area represents gingiva, and it is in this area that most of the time the biofilms are seen adhering to. Now, these disclosing agents are actually used in dentistry to educate the patients, because many a times the compliance on the patient side is that no, we brush the teeth. But then we need to educate them by telling them that no, this is how it is. Though it's not visible, but still the process is not complete. So that is one way of looking at it. If you see the photograph on the other side, you can see that the amount of biofilms formed is literally very, very insignificant. You can see them here in areas like spot-like areas, the coloration that has taken up. The entire thing is that biofilms form as soon as we brush, even after the brushing of the teeth, biofilm forms on the surface of the teeth by using the environment within the oral cavity to form a colony of microorganisms that sticks onto the surface of the tooth. Now, if you look at this clinical photographs, there is no inflammatory response seen in the mouth. That is, there is no gingivitis or periodontitis. There is no problem seen as such, though the biofilm forms this way. Because as I told you, as soon as the toothbrushing is over, also after some time, there is a formation of the biofilm. That is, our immune and other salivary factors have an important role to play in trying to prevent small amount of inflammatory changes in this particular environment. But as I said before, this pathology sets in when the microbiota changes because of certain changes that take place within the oral cavity. Now, these biofilms in the oral cavity can be classified as supragingival as well as subgingival. Now, as the term tells you, supragingival, you can see that the plaque is present on the surface of the tooth just beneath or below the area of the soft tissue, that is the gingiva. They are present below the gingiva. While subgingival plaques are hidden beneath the gingival tissues and the tooth surface. Now, it is likely that since the location is different, the organisms that are going to be present in these two locations are definitely going to be different, because the environment in these two areas are different, giving rise to a different colony of microorganisms in supragingival as well as subgingival biofilms. Now, when these biofilms are seen, it is evident that the gram-positive bacteria predominate in the supragingival biofilms, while gram-negative anaerobes are predominant in the subgingival biofilms. Now, as these biofilms keep forming into pathogenic plaques, it's going to become difficult because they cannot be removed by rinsing our oral cavity. They have to be taken out by mechanical methods like toothbrushing and other modalities, or in some circumstances, you need to take the dentist's treatment because the subgingival plaque is difficult to manage under normal circumstances, even though the hygiene is kept proper after a pathology has already set in. Now, I have tried to make this formation of the biofilm as simple as possible. This yellow area represents the tooth surface, that is the outermost surface of the tooth, that is the enamel. As soon as we brush after some time, there is a layer of organic component that forms on the surface of the tooth called as the acquired pellicle. Now, this acquired pellicle is a derivative of the component present within the saliva. So it comes and attaches itself on the surface of the tooth that is present within the oral cavity. In this situation, it's the enamel that is present under physiological conditions. Now, this acquired pellicle has receptors. These blue structures that have been shown represents receptors on the surface of the enamel, that is the pellicle that is present on the enamel surface, so that microorganisms can come and colonize. So in this way, different types of microbial species come and attach themselves to the acquired pellicle. Now, it is not possible for all organisms present in the oral cavity to attach themselves like this, because only those organisms that have cell surface adhesins that can bind to the receptors present in the pellicle can only attach themselves. And these first group of organisms that come and acquire the acquired pellicle via receptors represents the primary colonizers, following which the other organisms will come and colonize further, that is the secondary colonizers. Now, what dictates the secondary colonizers that are going to come and attach to the primary colonizer is that the microenvironment created by the primary colonizers is going to actually dictate the organisms that are going to be as secondary colonizers. And this addition of the secondary colonizers to the primary colonizers is called as co-adhesion. That means receptors should be present on the surface of the primary colonizers, through which the secondary colonizers are going to attach themselves and form a layer of biofilm. So it is seen that on the surface of the tooth, the organisms never attach themselves directly onto the surface of the enamel. In case the root is exposed, even on the surface of the root, it is the acquired pellicle that is first going to coat the enamel surface or any surface of the tooth that is exposed to the oral cavity. And then there is formation of a colony of organisms with time. Now, these organisms are going to use the microenvironment in our oral cavity, and they are going to live in harmony with each other. Now, this is not going to cause pathology as long as this particular microbiota is not allowed to extend further without disturbance. For example, as I told you before also, a poor oral hygiene. In case a person maintains a poor oral hygiene, this microbiota is going to change further. So basically, it is the organisms that are present within the oral cavity itself is going to give rise to pathology, provided the microbiological components within this change over time. So it is seen that the organisms are associated with each other. Many of them are going to be helpful, and they are going to allow or make the organisms thrive, and certain organisms which are not required are not going to be there, because there are certain substances released by these organisms which will only favor the growth of some required organism. Now here there is the same event here, that is the tooth surface. The yellow structure represents the acquired pellicle. Numerous receptors present on them. Around 60 to 80% of the primary colonizers are actually streptococcal species. Remaining are Neisseria, Haemophilus, Actinomyces species, Veillonella, and with time the other organisms as secondary colonizers start attaching themselves to the primary colonizers. It is seen that these primary colonizers usually take up the carbohydrates, and they are able to thrive in an oxygen environment. Facultative aerobes and aerobes grow. But with time, it is seen that anaerobes start growing as secondary colonizers, which attach themselves to the primary colonizers. And it is also seen that they use, instead of carbohydrates, peptides as a part of metabolism, for metabolizing or as a source of nutrition. They use peptides instead of carbohydrates. So a dense film consisting of numerous colonies of organisms are in tandem with each other, sticking onto the surface of the tooth. Now they give rise to so many pathologies, as I said. In this, you can see that there is a carious destructive process that has taken place on the surface of the tooth because of the biofilm. In this photograph, you can see that the gingiva is not normal. It is edematous, swollen, because there is a biofilm that has caused a change in this particular area, and it has modified the soft tissue component. This process is basically gingivitis, the initial inflammatory response of the gingiva to an exogenous component which is pathogenic in nature. With time, it is seen that this gingivitis progresses to periodontitis because tooth supporting structures, the alveolar bone and the periodontal ligament, are exposed to numerous toxins, enzymes, and various metabolic byproducts, and even inflammation is going to have an effect causing destruction, mobility of the tooth, and exfoliation at times. Or it can give rise to an abscess in the area of the periodontal ligament. So there is a wide range of pathologies that can set in when microorganisms are growing and they these issues. Now in this schematic representation, you can see that in areas where grooves and surface defects are there, these biofilms form very easily and they release acids in this conducive environment and cause demineralization of the tooth, and enzymes are released which cause dissolution of the organic component of the tooth. Now with time, it's going to involve enamel, dentin, and even the pulpal tissue. And if it is allowed to continue, it is going to affect the periodontal tissues in the apical region, and further causes of the sequelae of this process is going to start. If it is not affecting the hard tissues directly, there are other ways by which this pathology can set in. That is, here you can see one film of biofilm, that is the plaque that is formed here. This plaque is going to initiate an inflammatory process in gingiva. Where I said it is gingivitis.
Hello, ma'am. Can you hear us? Hello? Hello?
Tooth in such a way that tooth in this manner, wherein the tooth becomes mobile, and it may or may not. Madam, are you able to hear me?
Yes, ma'am, we can
Yes.
hear you.
So there are protective factors as well as pathological factors that are therein.
Can you please share your slides?
Ma'am, it is.
It is.
It's visible, madam.
No.
ಇಲ್ಲ ಮುಟ್ಟಿಲ್ಲ ಅಕ್ಕ, ಪ್ರತಿ ಸಲ ಕನೆಕ್ಷನ್ ಹೋದಾಗ ಸ್ಕ್ರೀನ್ ಶೇರ್ ಆಗು ಸ್ಟಾಪ್ ಆಗ್ತದೆ. Am I visible? Is the screen visible, madam?
Yes, ma'am. Yes.
I'm sorry for this. There's some issues of internet connection here. So these pathological factors can be because of a poor oral hygiene maintenance. Normally what happens is the process of demineralization and remineralization occurs on the surface of the tooth. That is, there are elements wherein the pathogenic organisms are going to release acids on the surface of the tooth. But we have inherent mechanism in the saliva to ward off this acidic environment, and there is no cavity forming in the oral cavity or no pathological event affecting the soft tissues, even if it occurs for the soft tissues. So there is a balance that is set in. But when this balance is broken, that's when the problem arises. So usually if you see the oral cavity on the enamel surface, basically it is dominated by non-mutant streptococci and actinomyces species. And the process of demineralization and remineralization occurs in a constant manner. But when there is a situation where the oral hygiene is not maintained or frequent uptake of karyogenic substances or too low a flow of saliva, then what happens is the remineralization is going to have a lower hold and demineralization is going to predominate. Now, in this scenario, the low pH non-mutant streptococci are going to reside in those areas, and with time, the Streptococcus mutans and lactobacilli are going to be present in abundant numbers and cause destruction of the tooth. Now, this Streptococcus mutans is able to thrive in an acidic environment, and they are also able to produce the acids that are going to act on the tooth surface, causing demineralization. So basically the organisms are the same, but the thing is that with time, there is a change of species of organism in such a way that only those organisms are allowed to remain, which can thrive in those areas as well as cause a destructive process. Now, I just got a schematic representation from one of the articles wherein this area represents the tooth and there is a pellicle formation here. Okay, and here there are receptors on the pellicle by which the Streptococcus mutans is going to attach itself so that it can start its pathogenic process. And we know that this Streptococcus mutans is able to synthesize glucans with which it is going to stick into onto the area of the tooth, and which serves also as its receptors and start a pathological process. Now, what should we do when this issue is there with us? The best way is mechanical plaque control measures using toothbrush, interdental brushes, dental floss in conjunction with fluoridated toothpaste. Now, the whole idea of using fluoridated toothpaste is that fluorides act on the surface of the tooth and form fluorapatite crystals. Now, these fluorapatite crystals, unlike the hydroxyapatite crystals that form enamel, dentin, cementum, they are going to be more resistant to the acids released by the organism. So this is one way of making the tooth a little resistant to the exogenous pathogenic substances released by the organism. But it won't decrease the acid produced by the organism. This we need to take care. Reducing dietary carbohydrate intake in the sense that there are many children or adults who have the habit of frequently eating food at very regular intervals. It is not that substances that are karyogenic has to be suppressed and we should not eat them. We can eat them during meal time before brushing our teeth. Instead, there are people who may not be eating highly karyogenic substance, but the frequency with which they eat can cause a problem with the ecology of the ecosystem of the oral cavity in terms of organism. And there are conventional methods of treatment when there is a pathology like dental caries, which has already started. So we know that there are many treatment options available of late for taking treatment for the same. I came across an article wherein they have used plant-based anti-karyogenic biofilm inhibitors. Now, these biofilm inhibitors work on basically three mechanisms. That is, they have an effect on the bacterial growth, or they prevent the adhesion of the microorganisms, the pathogenic organisms trying to adhere to the tooth surface, or they modulate the bacterial quarant sensing. Now, these quarant sensing molecules are small molecules that are synthesized. So there is probiotics can also be used and have been used to replace the pathogenic organism. And how they do is, the live organisms are administered in adequate quantity in such a way that they are going to house or actively use the nutrients present in the oral cavity for them to thrive, so that pathogenic organisms do not, or it is not made available to the pathogenic organism. Or another way that they do is by attaching themselves to receptors and making the pathogenic organism having no receptors to attach. And the most widely researched organism is the Lactobacillus species. And they have also done some clinical trials on incorporating these components in tablet, lozenges, and chewing gums that can be used easily by the person. As I told you, the mechanism of use of these probiotics is trying to prevent the growth and the attachment of these organisms in such a way that the organisms that are taken up as probiotics are allowed to grow rather than the pathogenic organism. Some of the probiotics the organisms in the probiotics are such that they synthesize antibacterial substances like bacteriocins that are going to be effective against pathogenic species that are present in the oral cavity, causing the pathology. So now if you see here in this tabular column, the probiotic is targeted against Streptococcus mutans and lactobacilli, and they integrate themselves into the biofilm. While there are others where they compete for the pathogens, or some of them will synthesize, as I said, bacteriocins as well as peroxide or organic acids that are going to have an effect on these karyogenic species. Nanoparticles have also been tried, wherein these small particles are enriched with calcium phosphate, and they have the advantage of having a larger surface area and targeting a particular localized area. And in addition to this calcium phosphate nanoparticles, they have also tried using quaternary ammonium salts as a combination of both antibacterial as well as remineralization properties. So this has been used. However, in our setup, we have probably, I'm not very sure whether in our country they are using these or studies have been done. Maybe it is in vitro that is probably tried rather than in vivo. But there are possible avenues where research can be done and can be made available so that it is beneficial to the humanity. Now, this oral biofilms, which is pathogenic, that is the plaque, can cause problems to the soft tissues in the form of gingivitis and then cause destruction of the tooth supporting structures, leading to periodontitis. So what happens is the plaque that is present in close proximity to the soft tissues is going to start an inflammatory response and release lot of enzymes, endotoxins, and antigens that are going to cause issues in the sense that they're going to cause destruction of the epithelium, connective tissue, and the supporting bone, leading to loss of tooth. Here in this clinical photograph, you can see that this person literally doesn't have teeth in the upper arch, especially in the anterior region. And if you look at his or her lower teeth, you can see that the root is also exposed to some extent. Now, this indicates that a situation of periodontitis has already set in this patient because of the poor oral hygiene that is seen in the patient. You can actually see the deposits on the tooth surface here. Other than being stained, there are deposits that are present, causing inflammation of the gingiva and the destructive process that it has initiated. There are methods of treating this periodontitis and gingivitis as well. The most trusted one being proper brushing and flossing. Here you can see a floss that is being used And here there is an incidental small brush that is used to cleanse the areas between the teeth. Now it is to be understood that the arrangement of the teeth and there are many other host factors that are going to dictate this accumulation of the plaque. Mouthwashes have always been used. Chlorhexidine is one common mouthwash that has been used in varying concentration, causing a change in the surface characteristics of the tooth so that microbial organisms don't colonize, that is pathogenic in nature. And there have been others that inhibit growth and colonization of organism. Now these plaques that form do not go if you rinse your mouth or gargle your mouth. So it is very important that mechanical methods of removal is very important, that can be done by us, preferably keeping the oral cavity clean, or seeking dental treatment where scaling, root planing is done, or if required, a surgical treatment is initiated. Now, I just came across while I was searching the information regarding new techniques wherein the biofilm can be altered. There are 4 mechanisms that were dealt. In one, there was modulation or modification of the enamel surface in such a way that it prevented the organisms from colonizing on the tooth. For example, silver nanoparticles were used on the surface to change its surface characteristics, which prevents attachment of the organism. Complete prevention may not be possible, but the number of organisms harboring to form a disease process is reduced. Likewise, antimicrobials have also been used in the past for the same. Other mechanism is that there is weakening of the biofilm that is formed so that the host immune system can act on it, or antimicrobials that have been used can be effective against these biofilms. Now, many of us know that the antimicrobials that we take may not be effective in the plaque that is already formed on the surface of the tooth, because the organism already has made a habitat in such a way that it prevents its entry, and it is seen that the concentration required to kill these organisms may be as high as 1,000 times that what we take to kill these organisms or alter the microbiota in the already formed plaque. So that can be one way of altering, that is trying to weaken this already existing ecology that is formed there. Now, one way of doing this is quorum sensing, that is trying to alter the molecules, the signaling molecules, such that they do not form a well-defined biofilm called plaque on the surface of the tooth. Now, there are other methods, that is the disruption. The disruption is in those areas where the plaque has already formed. So here enzymes have been used like DNases, where they target the plaque, the extracellular material, in such a way that it causes disruption and the organism does not have a surface area for attachment. As a result, the organisms do not house in those areas or cause the colonies to grow further. The last one is killing. Now, this conventional method have been used to kill the organisms from antimicrobials or antibiotics that have been used, and many other methods have been used before. Of late, they have even tried small electric currents that act on these biofilms, causing killing of the biofilms or even causing disruption of the biofilms. But this has to be tried on living patients, because something that is used in vitro may not work out in vivo, but the possibility always remains. Now, I conclude my talk by saying that dental biofilm is a where each colony behaves like an independent community, trying to thrive in its own environment, using the possible requirement or using the nutrition and other requirements for thriving. However, when the equilibrium is set in, there is no pathology. However, when there are environment is not conducive due to some predisposing factors, dental caries, gingivitis, periodontal diseases, abscesses, and many other sequelae can also crop up. Dental diseases are seldom exogenous in nature, that is, they are rarely caused by exogenous bacteria, especially when we know that biofilm associated dental issues are more than 85% of the issues that are seen in the oral cavity. So there is reorganization of this biofilm into pathogenic plaque where the composition of the biofilm is altered. And one more thing we need to know is the organisms that are pathogenic in our oral cavity can also have effect in our entire system because they can disseminate into the circulatory system or cause local invasion or enter the lymphatic system and affect the general health of the individual. There was a study done, I'll just tell a study that was done, wherein they have seen that a pregnant woman who has poor oral hygiene maintenance during her pregnancy is characterized by having babies of low birth weight. So the effect is so profound in trying to maintain our oral cavity is the answer for the problems that we see within and outside our oral cavity. Unfortunately, the current methods that are available may not always be successful in all patients, as there are inherent drawbacks which are patient related as well as with the system itself. So the aim is to identify agents or technology and methods that are useful for the patient without causing adverse effect and also helping the health of the patient. Educating patients in mechanical biofilm disruption is the main importance, and a change in lifestyle will definitely help in controlling biofilm related problems affecting the oral cavity. I hope this session was useful. Any question is there? Please be free to ask.
For an exhaustive presentation on oral biofilms. It was very informative. I'm sure the students would have really incorporated this because this is in the syllabus for the students also. I have a couple of questions here. What are your inputs to evaluate anti-biofilm assay on denture in patients who wear dentures?
Actually, madam, if you see dentures, no, I have not done any study on trying to find out what type of organisms are there. But in dentures also it is seen that when the denture is not kept clean, worn overnight or poorly kept or hygienic dentures, they tend to usually have candida organisms rather than bacterial organisms growing in them. So basically when you have such organism, the first thing is the organisms are more often seen in the denture area rather than the tissue area. That is one thing. So if you take organisms, take from the denture area rather than tissues, but tissues also show inflammatory changes. That is what I can help you in telling you about the organisms.
Another question is, can the overuse of immune system enhancers during pandemic trigger?
Madam, I can't hear you.
Can the overuse of immune system enhancers during pandemic trigger formation of more biofilms inside the mouth?
Actually, what happens, madam, when we use exogenous element, for example, if you take antimicrobials, if you are using, you cannot use it for a very long duration of time because they inherently have their own side effects. And these are going to have an effect on our system. Now, the thing is that, madam, when we use a broad spectrum antibiotics in the oral cavity for some reason for a pathology inside for a very long duration of time, it's actually going to change the microbiota of our system. So it's going to give pathogenic organisms to thrive. For example, you can person can get candida organisms. So that way. So long-term use of exogenous elements in enhancing or trying to reduce a biofilm is going to have an adverse effect also. Because as soon as you stop that exogenous element, it might give rise to a more prominent or excessive pathology or a denser film, like a thickened plaque than what it started with. Because there are studies which have been done where when the person stops that antimicrobial agent, it further enhances. Natural ones have been tried, madam. Antimicrobials or immune enhancers have been tried, provided they are not going to have a side effect for the same. But as far as the oral cavity is concerned, mechanical plaque, removal of these biofilms using toothbrush, interdental flossing, when done appropriately, are going to be 200% beneficial. The only issue happens with some patients who are having a system in such that they're quite resistant to some form of treatment. That's when we require intervention in terms of surgery or else.
We see a lot of advertisements in the media that use toothpaste, which kills 99.9% of germs, which we always see. And I don't believe all those advertisements. What's your take on that?
100%, madam. Don't believe all this stuff which says that it takes off the 99% organism, because organisms that are useful are also going to come off in the process, and we require organisms. The only thing is they should not house and alter the microbiota in such a way that they cause pathology. So we require microorganisms. We cannot live without them. Only thing is pathogenic organisms are prevented from colonizing. So you can actually even brush your teeth without toothpaste. That's not an issue. But actually it has lubricating effect. That is one thing. And another one thing that you should see is that better use fluoridated toothpaste. They use, in toothpaste 1,000 parts per million fluoride concentration, especially for children, because they are resistant to brushing and other techniques. Is going to be useful, especially when you know that the child is susceptible to infections like this. Dental care is because the child does not listen or is difficult to manage. And even fluoride applications can be done for children if they come to the dental clinic on a regular basis. That also is going to be useful.
Recording in progress. If this media is giving something like this kind of advertisement, why that Indian Dental Association has not raised any voice?
I really don't know, madam. Actually, I feel no doctor or anybody should endorse products that are given. There are people who are endorsing these products saying that they are good, but I don't know why they are not doing it, madam. They should actually.
What could be the receptors in biofilms formed on medical devices and environmental surfaces in hospitals, which are the common sources of nosocomial infections?
Madam, to be very frank, the receptors that are present on these structures, I have not done any study or evaluated because it doesn't come under my component. And being a pathologist, less of these are studied by us, to be very frank with you. So I am not aware of this. I wouldn't comment on this because it's not under my part.
There is a trend of putting tooth braces among some of the people. Will this cause any type of biofilm formation during or later in the later stages?
Actually, what happens now, madam, when orthodontic wire or any other treatment is put into the oral cavity, proper brushing is not possible. First thing is the patient develops intense pain and there can be ulcerations because of the inherent treatment and the force that is applied. Second thing is that oral hygiene measures are difficult in such patients because the wire is going to allow organisms in crevices to house there. But there are specific brushes available that can be used specifically by those areas. You can always ask the visiting orthodontist to give you the names of those that can be purchased in shops, and you can ask them how to use it. They are definitely going to help you. But inherent inflammation is very common in patients receiving orthodontic treatments. Especially these are done in childhood. And you know, no, madam, children, that's how it's very difficult.
Part of the human body. Are there any microbes which eats the enamel?
No, madam. Basically, microbes, what they do is, since enamel is 96% mineral, the mineral composition is as high as 96%. So what happens is these organisms are going to release acid as a metabolic byproduct, especially this sucrose-rich component. And this acid is going to act on the tooth and cause demineralization. It cannot eat up the tooth material. So this demineralization, when it is progressing and remineralization becomes less and less often, it is going to cause cavity formation. So all caries process need not start with cavity. Initially, there will be a white film that is formed. That is, the translucency of the enamel is altered initially. And then with time, as it goes on progressing, you can see small discontinuities in the enamel surface. With time, it becomes a big, large cavity, which cannot be treated other than intervention. The earlier process can be treated to some extent.
Are there any reports on oral microflora that can genetically inherited?
I'm not very sure about this, madam.
There is one more question here, madam. Does oral biofilms and alcoholics favor homogeneous microenvironment? How does it vary from the non-alcoholic individuals? And is there any particular species which are evolved to form biofilms and difficult to treat even in alcoholics?
Actually, ma'am, this alcohol uptake is there, no? It is even whether it is alcohol or whether it is cigarette smoking. There is a slight amount of change in the microbiota of people having these habits. Because basically alcohol causes dissolution of the epithelial component. They allow many substances to penetrate through them. If you see, this alcohol has this effect. And one more thing we should realize is that, madam, are basically people who are chronic alcoholics have inherently a decreased immune system. That is basically because the problem is general in nature. That is, people who are alcoholics, chronic alcoholics, have liver issues as well as nutritional deficiency. And this is actually going to harm the person in altering the immune mechanism of the person. So it does have probably a change in the microenvironment. And other than that, probably these alcoholics also might not be able to maintain oral hygiene also for the habits that they have. That could be another reason.
How effective is the traditional method like oil pulling effective in eliminating oral biofilms?
Oil?
Oil
What is
pulling. That is rinsing the mouth with oil.
Oh, no. We have not encouraged anybody to do like that. It is not going to have any effect, madam, to be very frank, trying to take oil into the oral cavity. Because these biofilms, even after a biofilm has been formed, we gargle our mouth, still the biofilm persists. So just putting oil into our oral cavity, it's not going to work out because this oil is going to be swallowed by us only afterwards. Even if you rinse it, the amount of oil that is going to be present in the oral cavity is going to enter our system with the organism. So it is not going to have any effect. It might not last very long. So these are not solutions to trying to control biofilms by using oil.
One more question. Which time is more appropriate to brush? I suppose it must be.
Madam, I can't hear you.
Which time is more appropriate to brush? After meal or before meal? Morning or night?
Actually, to be very frank, I brush my teeth before my breakfast. After my breakfast, not before, after my breakfast. The thing is that, madam, once we eat our dinner, we brush our teeth. That is appropriate, 100% fine. After that, we sleep off and we get up, but there is no substrate for the organism to work on. So there is no point in actually brushing our teeth when there is no substrate given to the organism to act on, because we assume that our oral cavity has been kept clean by proper brushing, and that removes most biofilms, but though it forms again, they are not going to be pathogenic to us. So preferably it is better to brush the teeth after you have breakfast, because that helps in controlling the formation of these pathogenic biofilms. But many of them might not tell you this, but I'm doing it for myself, my son, and others also. Yes, madam. I can't hear.
Yeah, one more.
What
What is
is
the charcoal in toothpaste in the biofilm formation?
Madam, I
Safe
don't.
to use charcoal?
Actually, they are just like abrasives, you know, madam. I don't. Probably they are methods to promote a particular commodity. To be very frank, the time taken by brushing by an individual is actually less than 1 minute. The time taken that we take time to brush. But though the recommended time is minimum 1 to 2 minutes, that much time we do not spend. And using these charcoal and other elements for brushing is of no use, provided the proper technique is to be used. Whether you use a Colgate toothpaste or a Patanjali toothpaste, if your method of brushing is wrong, no matter what you add is never going to remove that material. So it's a technique that is useful, not the material with which you are brushing is going to be useful. And there are reports wherein plain toothbrushing is also without a toothpaste can also remove plaque material. But I feel using toothpaste is going to be more effective, and it has that lubricating effect, because when we brush without toothpaste, it has a different sensation altogether, a little unpleasant one, I should say.
Now, can you take one more question?
No problem, madam. Please ask.
Okay, so what is the reason for throat stone formation? And
Pardon
what type
me?
of throat stone formation? I don't know whether it is strep throat or throat stone.
Throat stone formation.
No, I don't know what is throat stone formation.
Okay, ma'am, can you tell us something about strep throat and what type of oral hygiene should we maintain?
Preferably, they say that use a soft bristle toothbrush, because it's going to be detrimental to the soft tissues that is there. And the soft, but I don't use soft bristle because it for me it doesn't work out. I feel a medium bristle toothbrush is going to be better. So when you're using a toothbrush, you should see that you change your toothbrush once in 3 months. It is not that a toothbrush is just taken and the end of 3 months you're going to throw it. It's not based on that. The fraying of the bristles will start moving outwards with time. In case your bristles fray out earlier before 3 months, that means you are applying unwanted pressure on your teeth and causing problems. With time, you will end up causing gingival recession and other problems. If they don't fray out within a span of 3 to 3.5, 4 weeks, months, that means you are not applying any pressure. The pressure applied is very, very less. So that is optimum pressure, and brushing has to be done for upper teeth from upwards to downwards, not opposite. Because if you're going to do opposite, then the tissues, the soft tissues are going to move out, and your, you'll get inherent problems of sensitivity. And lower teeth also has to be brushed from downwards to upwards, and upper teeth from upwards to downwards. All surfaces of areas of the tooth you rinse once you brush it, rinse mouth after every meal or anything that you have taken. And brushing thrice is okay in case you realize that your child or anybody whom you are seeing is having a ability to develop caries very soon. You can always, and please recommend children to take fluoride application on their mouth when they are small, because it's very difficult to manage children of late, because the amount of processed food that is available to attack the teeth is numerous.
Thank you so much, madam, for your insightful talk. It has been extremely informative for all of us students. So we really thank you for your time and taking part in this webinar.
Thank you from my side also, madam.
we have come to the end of this webinar, and I would just like to propose a word of thanks. So I, on behalf of all the SIG members in the group, which is Dr. Jamuna, Dr. Sindhu, Dr. Sumana MN, Dr. Durai Anand, and Dr. Mahesh, on this occasion of international webinar on clinical biofilms and clinical settings, would like to take this opportunity to thank JSS Academy of Higher Education Research for sanctioning funds to carry out this research and this webinar session also. And a special thanks to all the three speakers, Dr. Devendra Dusane, Dr. Vandan Nagar, Dr. Veda Hegde, for taking time and sharing the rich research expertise in the area of biofilms. I thank our HOD, Professor Ravisha KA, for their support. And I thank the participants, importantly the participants, for joining the webinar and having asked a lot of questions. I'm sure they have imbibed the knowledge what they have learned today. And I thank the information technology section, Dr. Ravindra, for helping organize this webinar online. And I finally thank the leadership, our co-chancellor, Dr. B. Suresh, our vice chancellor, Dr. Surendra Singh, our registrar, Dr. Manjunatha, and our director of academics, Dr. Kushalappa, for the constant support and encouragement. This webinar is officially now over. Thank you, madam, for your inputs, for your time. Thank you.